Hello. Hello, good morning to all. So I also take this opportunity to thank the organizers and particularly Dr. Ashok Bandera from Amity Institute of Food Technology, who has given me this opportunity to speak before all of you. So it is a pleasure. The topic that I've chosen for this presentation, edible desert legumes as sustainable and healthy foods. So I'm coming from WITS Pilani campus. So we are working on the plants which are present in plenty in the desert regions. So I will elucidate some of our research work that we have been doing. So now all of us are aware that sustainable development goals put forward by the United Nations. These goals are telling us to produce food for the growing human populations in a sustainable manner, because we have to take care of our environment also. At the same time, the human population is increasing. So with the current food production methods, which are also contributing to pollution, we may not be able to cater in a safe and sustainable manner to feed the hungry population of the world. So here we have to target both malnutrition as well as overnutrition, because all of us are aware that many diseases are also happening when we are over feeding ourselves. So I was visiting one farmer in Himachal Pradesh. So it was like busy in the fields, plowing the fields, and after working in the field during the lunchtime, one big slice of butter he put on his plate. And I said that with me also. He said that you will not be able to digest it because your lifestyle is not that physically demanding. So when we talk about our food, it has to be balanced with proper lifestyle also, where by exercise, we are also spending the energy that we have taken. So we have to take care of the sustainable development and see that the food products that we are eating and not only feed the target the hunger but also the overnutrition. So one very interesting model was given by some scientists, environmental nutrition model. So they say that when we are talking about food for the humans, nutrition has to be there. At the same time, health has to be there. That is both nutrition as well as health benefiting phytochemicals or nutraceuticals have to come from the food. Nutrition plus nutraceuticals. At the same time, the food has to be prepared in environmentally sustainable manner. Now, right from farming till the time the food reaches the customer's palate, there are so many avenues for greenhouse gas generation. For example, fertilizer application, fertilizer production, then transport. Okay. So this environmental nutrition model says that it would be good if we can have data for all the major food crops. If we can have data for all the major food crops, where we know that what is the environmental burden created by producing this particular food. So this data, if you try to see environmental nutrition model for the different food crops, are they available or not? We won't find for many crops. For example, for the major crops, wheat, it may be there for wheat. But for the lesser known plants, lesser result plants, which we are consuming, but that kind of data is not there. That is to produce say 1 kg of food from that particular plant. What has been the environmental burden which has been created? For example, in terms of greenhouse gas generation emission. So I often like to travel, and sometimes we make a tour in Rajasthan, sometimes Himachal Pradesh, sometimes near the seaside. That's the beauty of our country. So we are blessed with a huge diversity. And when we go in the deserts, particularly now also we see that the temperature is rising. In winter, it may be okay. But for people like us in winter also, it is too much cold weather and not able to hold it. But in the hot desert conditions, we see that what is there, perhaps oasis will be there, some small pool of water. We will see some camels. But the desert is hiding a lot of treasure. Sometimes in the fictional stories of Aladdin, etc., we hear that there are treasure in the deserts. But the treasure that I am talking about are the plants, desert plants, because these desert plants are exposed to very harsh environmental conditions. For example, here also Delhi climate is also similar. So we will have 0 degree in the winter and 48, 50 degree in the summers. Okay. So to take care of this extreme of environmental conditions, the temperature, the harsh environment which these plants are exposed to, the plants, they cannot run like animals, they cannot escape to a different place. These plants, they accumulate very unique and diverse and distinct metabolites. These metabolites are accumulated by the plants in response to stress. Different stresses that they are getting exposed to, for example, high temperature, low water conditions. And these same metabolites, when we consume through the food, they are acting as food. These are different metabolites in the plant, but we are treating them as nutraceutical. So this means that the plants in the deserts, they are harboring some unique, special kind of metabolites, which are actually the treasures, which we can use for the medicinal and pharmaceutical or nutraceutical purposes. The scope of the desert plants, particularly I will be talking on some of the underutilized desert plants. So we have been dependent only a few food crops like wheat and rice. But I ask you that, is it wise to depend only on a few food crops? It is not wise, because suppose the climate is changing suddenly or there is a pathogen, new pathogen attack, then the monoculture which many of the farmers seem to be going today, so all these crops might be decimated. So we have to look at some of the underutilized plants also, because we have to harness the diversity which India is blessed with. So these underutilized plants, they are well adapted to the extreme environmental conditions. No inputs are needed in terms of, say, fertilizer, in terms of water. They are already acclimatized in low water conditions, in drought conditions, high temperatures, low temperatures. And best of all, we have to look for those kind of food giving plants which are local, reasonable, and sustainable. Because let us connect back to the environmental nutrition model that we're speaking about. If there is a local plant, food plant, then the local communities who are living near the plants, they will not require too much of transport. Okay, unless and until we are processing the food product deriving from that plant and exporting somewhere else. Okay, so locally grown fruits and vegetables, they are better, because there will be less application of fertilizer, etc. So we have to go for local, seasonal, sustainable. Now some of the legumes of the Indian desert, can be removed. Might have heard of this special dish from Rajasthan, Kair Sangri. Have you heard? Special dish, Kair Sangri. So this is the Sangri, Propopis senior area. These are the Sangri pots which are used by the Agri tree. And Prosopis senior area, it is also the state tree of Rajasthan. State tree of Rajasthan. And all its parts are crystal. Then Cymopsis tetragonolova, this is guar. Although this has been a good commercial source for the food stabilizer guar gum, but with respect to the nutraceutical and antioxidants, it is somewhat less request. Then there is Acacia senegal, which is seeds which are obtained from the pods. So just like Kair Sangri, there is a special cuisine in Rajasthan that is Panchputani sabji, we have heard. So these Kumatia seeds are added there. So some of these plants, many of these plants, either they are constitutively accumulating the different metabolites, secondary metabolites, or as a response to abiotic and biotic stresses. You might have heard of that story, that the Bishnois, they protected the HD trees when the people of the king, they came to cut the trees because the king wanted to find his palace. So he needed bricks to be heated up in the field. For the firewood, he asked his men to go and cut the HD trees to bring back the firewood. But the local people there, particularly the women, they started cutting the trees. You might have heard of the tree. So it started from here. And I always like to imagine that at the end of the day, when the nomads, they are planted to the harsh desert conditions, the deserts of Rajasthan. Then at the end of the day, they are halting at some place, getting camp, and eating something nutritious. So these kind of things which our local communities, they have been dependent on for a long time. Now it is time to harness them to produce perhaps proper products, okay, good nutraceuticals, healthy nutraceuticals from these kind of plants, for which already the traditional validation has been done to also identify some of these nomadic communities. So talking about the Prophytis cinerea and Samopsis tetragononova, traditionally also people have been using them in ethnomedicine. So they have been reported for various diseases, for example, blood purification, skin disease, respiratory problems, anorexia, arthritis, etc. But what the scientists are looking for is today to find out the mechanism how these could be acting in all these kinds of diseases. But what the point that I want to convey is that although these plants have been used traditionally, but a scientific validation and a scientifically validated food product might not be still present in the market like we have for wheat, rice, corn, etc. So here we did some research on this Prophytis cinerea pods, and we harvested them. Took the pods from different regions in Rajasthan. Now you know that Rajasthan, there is arid region and then there is semi-arid region, which is divided by the Aravalli Hills. You have heard of Aravalli Hills? So both on this side of the hill, there is the rain is somewhat more, although nowadays the climate seems to be changing a little. Arid regions we have no rain for semi-arid regions. We are living in extremely changing climatic conditions. But what we did here was we took the edible pods from the different regions, and we saw that the total phenolic content in all these pods from different regions, the trees from different regions, they vary a lot. This means if we are trying to develop a nutraceutical product from these trees, then we have to take care of the fact that region-wise variation can be there. So those particular germ classes or accessions have to be selected. So then we went about purifying these metabolites. So we wanted to enrich the enriched fractions. So there is one very good article on obtaining flavonoid enriched fractions from honey. So we use that method where the amberlite hexane and Cephadets LH20 chromatography was used, and ultimately we could obtain flavonoid enriched fractions, and after LCMS analysis we could observe the number of flavonoids and isoflavones. So why I talk about isoflavones? Because the legumes are rich full of isoflavonoids. Soybean being the most well-researched plant. So in Japan, they say that they are having less of cancer, less of estrogen hormone related problems. This is because they are taking a lot of purify the compound, because mechanism, for example, we want to see that what receptor that compound is binding to, which receptor on the cell, then which genes are overexpressed after that metabolite binds to the target receptor, which are underexpressed, downregulated. But still, for the activity purpose, rather than going for the pure compound, we may like to have crude extract. So here we have did some research on deriving antioxidant activity enhancers from this guar pods, Samopsis tetragononova. So we have heard of antioxidant activity in these plants, but here we have used them for enhancing the extract obtained from the guar, for enhancing the antioxidant activity of some known dietary phytochemicals. So we chose 5 flavonoids and 5 non-flavonoid phenolics. And we saw that at a particular concentration of the pure phytochemical, as well as the particular concentration of the guar seed derived extract, there was synergistic effect on the antioxidant activity. And that antioxidant activity we also verified through cell cultures. So we created oxidative stress model in fibroblast cell cultures and saw that the kind of results, antioxidant activity enhancement that we saw in the cell-free extracts, we could also see in the cell culture models of oxidative stress. So the hydrogen peroxide derived oxidative stress was reduced. Now the question that I was dwelling upon a few minutes back, to purify or not to purify, because as I said that if you want to study the mechanism, you should purify the pure compound. But overall biological activity may not be because of only that one compound. It may be because of a mixture of the compounds which are acting synergistically. For example, here, if we look at the crude Cymoxis tetragonolaba guar crude extract, its IC50 value is 7.25. But when we enriched the flavonoids, it was higher. So we see that this crude extract is having a better antioxidant activity. Although I would like to stress here that high antioxidant activity may not be the only mechanism when we are looking at the disease preventing or protection from the diseases. The mechanisms might be something else also, but here we are looking at the antioxidant effects. And we have also done some metabolic profiling from these desert plants. So you know that these metabolites which are produced by the plants as a defense response, they may be toxic to the plants themselves if they are allowed to stay there in the long run. So what the plants do, they glycosylate these metabolites, add sugars, and make them less toxic and more soluble. And these glycosides are then stored in the vacuoles. And when needed, rather than producing the toxic metabolites, when needed means in times of stress. For example, when herbivore is attacking, when pathogen is attacking, or when there is high temperature stress. So instead of producing the metabolite from scratch, the plant recruits one enzyme, beta-glucosidase, which will cleave the glycoside, which is stored in the vacuole, and release the pure A-glycon, which is often more bioactive. And we saw that here, these enzymes, the glucosidases, they were very specific for the isoflavone glycosides and not they could not act on the other flavonoid glycosides. So these are very specific enzymes. So some of the challenges and opportunities, I have told you that these desert plants are source. They are the treasure trove of bioactive molecules, but there are some challenges associated with them also. So this is the Sangri pod. Okay, Sangri pod, but what happens that if proper heat is not there, for example, we have been experiencing also sudden rains and the temperature going down in March and April. So what happens, instead of the edible pods, this kind of gall develops, okay, in those pods. So they are no longer edible to the humans. So this is one challenge, some of these diseases. And the yield wise also, the kind of germplasms which are present, their yield is low. And of course, say when we are fond of rajma, chola, but this is also legume, but its taste, if we are eating them regularly, we may not like the taste as much as rajma and chola. This means there is scope for improving the organoleptic properties, taste, texture. Toxicity studies have to be done because whenever we are talking about deriving nutraceuticals and consuming them, they are enriched products. So if they exceed the particular dosage, they might become toxic. Processing can be done to reduce the anti-nutrients, which might be giving some off flavor to these foods derived from these plants. Economic evaluation has to be done. How well the farmers are able to grow these if they are supplying these pods to the industries, food processing industries. Okay, how they are able to take care of the diseases, what is the yield. And once we link these kind of plants to the industries, there will be opportunity for the local growers also, but for the same, these challenges have to be overcome. So just one hypothetical idea we gave recently in one article. So we have said that what's good in these local legumes, they are accumulating some unique bioactive metabolites. They are highly stress tolerant plants. Okay, they can tolerate extreme climatic conditions, which we are facing now. And they are environmentally sustainable because they require less of water and fertilizers and pesticides. But what's not so good? Okay, beanie astringent off flavor, because of which they are not so popular. So we take the Kher Sangri once in a while. Okay, only as a special dish, but we are not taking them regularly. Anti-nutrients have to be taken care of. Availability may be only for a particular period of time. Yield is low. So we have suggested that by CRISPR-Cas, perhaps targeted genome editing can be done to take care of these not so good factors present in these plants. In conclusion, I would like to say that many of these traditional edible plants, their usage has been validated through the ages because the local communities have been using them. for food, ethnomedicine, for fodder for the animals, for firewood, etc. So when I told you about Prosopis cinerea, khejdi, which is the state tree of Rajasthan, all its parts are useful. Leaves are useful for the cattle, pods are useful to the humans, bark is useful for the skin ailments, and being the legume plant, they're accumulating nitrogen fixers. So if functional foods and nutraceuticals can be developed from these under-researched or neglected legumes with minimum inputs from the environment, that is in terms of water, fertilizers, pesticides, etc. In the end, I would like to acknowledge the organizers, participants of AMIFOST 2023, particularly Dr. Ashok, who gave me this opportunity, administration of BITS Pilani, SERB New Delhi, and all my students, because I teach different courses, some courses related to food, some courses in biochemistry, and the classes become enriched when the students interact. So our joy, I find that in the 24-hour period in our profession, what is the most joyful time? That is when we are interacting with the students. So I'm thankful to my students. And this is my group, research group at BITS Pilani. So myself, along with Professor P.R. Deepa, she's also a biochemist, and our students, Tripti Joshi, Sumit Mandal, Sonakshi Puri, Vidushi Asati. So we are blessed to have very hardworking students. So it is all a teamwork. Thank you very much. I would like to reinforce that to eat is a necessity, but to eat intelligently is an art as well as a science. Thanks to all of you. So if there is any question, I would be happy to answer.
Thank you, Dr. Pankaj Sharma. And now session is open for any discussion. If you have specific question too. Congratulations, Dr. Pankaj Sharma, for a nice presentation.
Thank you, sir.
I have one query here. Have you ever gone through any literature, or have you ever worked on the toxic compounds present in these desert plants?
Toxic compounds, sometimes they are accumulating these saponins, which at a very high concentration, they can be toxic. And obviously all these metabolites that I am talking about, phenolics and flavonoids. So if we are doing biological activity studies with them, we see that at a higher dosage, they are toxic also. But it is a matter of dosage. The same compound, when medicine at a lower dosage, at high concentration, it will become toxic. So this toxicity factor has to be considered.
Is there any work going on in India where they are working on it to reduce these toxic compounds?
Toxic compounds, as I said, that those kind of germ plasms, the scientists are trying to develop where the toxic compounds will be less, the anti-nutrients will be less. So the work is going on in, for example, the ICAR Bikaner, then in Central Arid Zone Research Institute in Jodhpur. So they are working on these plants, also toxicity related aspects.
Okay, thank you.
Thank you. Thank you.
Thank you, Dr. Sharma, for such a wonderful talk. So we have next invited speaker, Dr. Pradeep Kumar Singh. Dr. Pradeep Kumar Singh is an associate professor in Veterinary University, Jabalpur. And today he will deliver a talk on focus on nutritional security and health with functional livestock products. So we welcome Dr. Pradeep Kumar Singh, please. जी कोई इसके अंदर वो तो नहीं. Thank you, Dr. Ashok Pathera and the team Amity here. A very good morning to all the distinguished scientific fraternity present here. So, my topic of discussion today is how we can focus on the nutritional security and the health aspect to the functional products that are derived from the livestock origin. Since I'm a man of LPT dealing in the livestock products, so I'll focus my discussion towards the livestock products only. Although people from the different scientific diaspora here, so naturally they will come to know something different from the livestock products. So as we know, initially the country was starving, and although still the reports are there that people are starving in different parts of the country, but more or less majority of the population, at least they have got a food today. So now the time is, since we have quantified, now we have to move towards the quality food that is nutrient rich, or that has some component that is altering the biological activity or enhancing some physiological benefits to the human population. So that is why we are moving towards the functional food products. And the problem is more aggravated with the change in lifestyle, as we people are living in urban areas, and majority of the time we don't have time for processing and all these activities, and due to lifestyle, due to low exercise and other physical activities, majority of are suffering from this diabetes or some cardiovascular ailments or hypertension and other types of lifestyle diseases, and majority of them, they are of metabolic in nature. So there comes the role of the food actually. You need not to go for the medicine always, just like the diabetes. Majority of the people are tackling diabetes just by the changing their lifestyle or changing their food habit. So similarly, other diseases can also be tackled by the food habit only. As we know, food has got all the components or elements, and some of them they are beneficial to us, and some of them they may be deleterious, having some deleterious effect. So we have to enhance all those components that are having some benefits to the human body, and we have to actually reduce the components that are causing some ailments, some problems, some toxicity in the body. Or suppose if a food is having high salt content, then you need to reduce the salt content for the hypertension peoples. Or similarly, for diabetic person, you need to reduce the sugar. So naturally, that is why low sugar products are developed and they are available in the market. As we know, majority of the if we call the quality protein, animal livestock products has got a very major role, because all the essential amino acids and fatty acids, they are present in the meat and other livestock products. So naturally, people are preferring for the protein source or high quality fat source. We have got a very traditional history where several Ayurveda treatment and other practices, they were followed in India, and somehow they have got some dysfunctional aspect. They are enhancing some of the components in the either in a herbal product or some other milk based products. So what are the products that actually qualify for the this functional products? So it should not be a capsule or a tablet powder. It should have to be a natural ingredient and should be a part of natural food habit, and it should have some functional property. That means if you are taking for a purpose, for example, if you are suffering from some ailments,
like if a person is suffering from this diabetes, so naturally he will not go for the sugar. He will go for some alternative sources or alternative sweeteners. So that is incorporated in those products. That is also a type of functional products only. This is just to show the lifestyle and the ailments we are dealing today: obesity, heart problems, cancer, diabetes, osteoporosis. There is plethora of diseases that we are actually facing today. So this is a little bit information about what is the status of livestock product availability in India. And as we know, India continues to be the largest producer with around 210 metric ton of million metric ton of milk production, and per capita availability of 427. Similarly, the egg availability of 122 billion and meat 8.8. But as we know, since an adult requires at least 56 grams of protein per day, which is RDA. So, and government is planning to have some at least 50% of the source from the animal from the animal sources like milk, egg, or meat. Majority, the government is planning to enhance the production of fish production or the meat production, so that at least you should get the 28 or 29 grams of protein per day from the animal source. Now, these are some of the functionalities that actually carry the livestock products. For example, if it is a dairy-based products, so naturally it is having a very good quality proteins, whey proteins and whey peptides. Similarly, the polyunsaturated fatty acids from the fish, especially the omega-1, and conjugated linoleic acid, especially from the ruminant, either it is milk or meat, and sphingolipids from eggs and some plant sources also. And similarly, meat is recently has been found to be having a very important source of compounds like carnitine, lipoic acid, and anserin, and other components. Carnitine is a very good energy source that is actually required by the people who are exercising heavily, or this pregnant ladies or lactating mother, where the energy requirement suddenly enhances. There you need to supplement, and this works through the beta oxidation. This is the omega-3 fatty acid class of especially the EPA and DHA. There are two mainly. Now, if we come to the history, we'll find that long back, this omega-6 and omega-3 ratio was 1:1. Then later on it changed to 10:1. And now the figure is quite, which should not be there, 20:1. Now, what is recommendation? Recommendation of this is it should be between 1 and 4. This ratio should be between 1 and 4, not more than that. Regarding this saturated and polyunsaturated fatty acids, that ratio should also be between 0.4 and 1. As we can see, it is only 8 to 12% of the recommended daily intake we are taking in present day scenario. And regarding this omega-6, it is almost more than 1,000 to 2,000% of the recommendation that we are taking in the food habit. And if you have to enrich your this omega-3, then you have to certainly go for the marine sources like salmon, tuna, mackerel, and other fishes, so that you have enhanced this omega-3 to balance that unbalance that I have just shown in here in my previous slide. And some work is also going on to enrich this omega-3, and some products are also available in foreign countries like Canada and USA, where they have enriched this omega-3 meat and they are marketing it. This is just a classification of fatty acids. Again, this omega-6, linoleic acid, arachidonic acid, and omega-3, alpha-linoleic, EPA, and DHA. The different sources, majority of them, they are sources fish oil or the animal fat. Now, CLA, as we know, it's a mixture of positional isomers, and majority of them, they fall under the 9 and 11 and 10 and 12 categories. The major source of CLA is the ruminant fat, that especially the meat or the milk. suppose we are going for this goat meat or sheep meat or this buffalo meat. So they are very good source of CLA, because bio hydrogenation occurs in the rumen. So that is why during that hydrogenation process, these compounds are formed. Cow milk fat is the richest natural source, and ranging from 2 to 37 milligram per gram of fat, and beef fat that also contains 31.1 to 8.5 milligram per gram of the CLA, and the other one is similarly the other functional compounds is meat, or is a very good source of protein. So naturally the bioactive peptides will obtain from it. It will be of a very good quality. So and we all are working, and we all are reviewing the papers, and we are finding a number of this function for the bioactive peptides, and they can be produced by food processing methods like treatment of enzymes and some other fermentation and some other techniques. So there are several functions like this antioxidative activity, anti-thrombotic activity, anti-hypertensive activity, immunomodulatory activity, and several functions that are claimed today through these and this bioactive peptides. Coming to the probiotic food, milk is a very good carrier of probiotic. So naturally, it is one of the most preferred. Although some meat products have also been developed as a probiotic meat products, especially the sausages, fermented sausages, they have been used as a carrier for these probiotic organisms. And we all know about the nature of probiotic. Just like bioactive peptides, these probiotics have got a number of beneficial effects to the body, like they are anti-mutagenic, anti-carcinogenic, antidiarrheal properties. Then several these pathogens, they are inhibited when they are present in the GI tract. Then coming to the next important food component, that is sphingolipids, and major source is the egg. Although some other sources they can also be taken as a sphingolipid source, and it is indicated for the cardiovascular diseases. It reduces the risk of CVD. Next is L-carnitine. It has been claimed as a magic compound today because it is a very good energy booster. People who are exercising heavily, or these pregnant ladies or lactating mother, those who need sudden enhancement in the energy, they can go for this compound, and it is marketed. It works through the beta oxidation chain and enhances the energy production. There is a recommendation also that you can take it from 24 to 81 milligram per day. Then alpha lipoic acid, then carnosine, anserin, they all have got antioxidant activities, and they have been used in other products also to enhance that activity and to enhance the storage qualities. So there are several strategies actually to develop these livestock products. One could be to enhance the beneficial components, or otherwise to reduce the components that are having some ill effect. One could be the genetic level, because as we know, the golden rice and other products have also been produced to enrich the vitamin A. Similarly, people are working to produce such birds or animals that can lay eggs with enhanced vitamin E and vitamin A. Something like that, people are already working on it. And next is the feeding practices. You can just give the enriched feed with the vitamin A, selenium, and other components that will be reflected in the product, either it is milk or meat or egg. And they have been even produced and marketed. For example, this omega-3 enriched eggs, they have already been produced, and this flaxseed have been used to enrich the omega-3 in some of the meat. Then supplementation with selenium and magnesium and or iron, that has also been employed to enhance these products in the meat. Similarly, the DHA or EPA, they can be enhanced or 7 to 8 times, something like these findings have been reported. Another practice is to incorporate in the functional, incorporate these functional ingredients or modify the product component. No, incorporation is simply you have to include in your formulation. The formulation you are preparing, simply you have to add it to the product directly. Then some modifications. Suppose if a meat product is having higher saturated fatty acids, then simply you have to remove. If it is a removable fat, just like pork, you can remove that fat, or chicken, where there is subcut fat and fat adhered to the animal tissues, that can be simply removed, and you can add the polyunsaturated or monounsaturated fatty acids as per the requirement. Similarly, CLA and other components that can also be added. Now, some practices to reduce the cholesterol level, they have been employed by using organisms, and some chemical treatments and other physical processes they have been adopted, like NDRI is producing low cholesterol ghee. So there are people are working on it also. Low calorie products, naturally you have to reduce the fat content in the product. Similarly, the low sodium product for the hypertension people. Nitrates and nitrites that has been reduced by incorporating some plant ingredients, especially beetroot and other components, so that that color can be developed in the meat products, because majority of the time this nitrate or nitrite is used for the color development. Now, some challenges like safety. Once you are incorporating something, you have to be very much sure about the safety of the product, how much safer it is going to be if it is to be taken from the other sources, plant sources or some other sources. And naturally, if you are altering the food formulation and food component, then sensory and physicochemical properties, they are compromised there. And some interactions will be there. That interaction needs to be studied, because majority of the time what we are doing, we are simply formulating a product, but we don't go for the interactional study. That component is interacting with the other components also. And higher price, because once you will go to the market, if you will ask for a low sugar sweets, so just simply the price becomes 1.5 times or 2 times. The 400 per kg that burfi becomes the 600 or 700 or 800 per kg, although sugar is not used there. So price is also one of the limitation. So to conclude my discussion today, we need to work a lot on this, because it has got a number of benefits, and those benefits are related to the lifestyle diseases, and we are majorly suffering in today's scenario. So we have to work on it and develop some products like that, so that though the challenges, they are ruled out, and we come out with a product that is readily acceptable and having all those beneficial effects to the population. Thank you.
Thank you, Dr. Singh. And the session is open for a short discussion, if you have any.
Hello, Dr. Singh. It was a nice talk.
Thank you, sir.
And you have talked about the good quality protein from animal products, animal meat, for example. So for vegetarians, what is your suggestion?
This is my personal view. Majority of the vegetarian, they used to take the milk and milk products. So naturally, they are taking the good quality protein through either paneer and other type of products. So if a person is vegan, then we need to concern about that. Otherwise, meat is not that much included in our day-to-day diet. Milk is a part of, from milk to dessert, we are taking milk throughout the day. So we need not to concern about that.
Milk to dessert and mainly tea.
Tea.
If we are adding enough quantity of milk. So yeah, for vegans, it may be an issue. So we have to look for good sources of good quality proteins with all the essential amino acids. That is an area of research. Thank you.
Thank you, Dr. Pradeep Singh, for such a wonderful talk. Thank you.
Thank you. Thank you.
Now our next speaker is Dr. Ravindra Kumar. So Dr. Ravindra Kumar is currently Assistant Professor in Guru Jambheshwar University of Science and Technology, Hisar, Haryana, and Department of Biotechnology. Dr. Ravinder has done a extensive work on the aloe vera, and today he will speak on aloe vera, a universal panacea to the health culprit. So we will welcome Dr. Ravinder, please.
Good morning, respected Dr. Abdul Rashid, Professor P.K. Singh, Dr. Neeraj, Dr. Pankaj Sharma, and all the other participants. I welcome you all. I just want to highlight some dark side of aloe vera. As we are consuming lot of aloe vera, and you can see there are different type of beautifying products we are using. And before these researches, we people only think that the aloe vera is a universal panacea. It is a universal solution. Every skin elements, any health problem related to stomach, that can be corrected by consuming aloe vera. So my topic is aloe vera, a universal panacea to a health culprit. So as we can see, from the traditional system, we are just relying on the medicinal plants. Actually, 60% of the Indian population still relying on the medicinal plants. And in case of food, medicines, cosmetics, we are consuming a lot of medicinal plants. Then if it comes to aloe vera, aloe, that means a shining bitter substance. Vera, that is true. And some Greek researchers, they called aloe vera is a universal panacea, is a universal solution. And Egyptian called the plants of immortality. Then in India, it is commonly known as Grit Kumari, Gawarpatha, etc. And this plant is basically indigenous to South Africa or the Mediterranean countries. If you talk about the total species, then over 530 species of aloe vera are present worldwide. But among them, there are 4 to 5 genotype or species, that is aloe perry, aloe barbadensis Miller, aloe arborescens, aloe vera. So among them, aloe vera is the most potent plant that have the maximum medicinal property. So among 530 species, only 4 to 5 species are potent, which have good medicinal properties. Then it is a xerophytic plants. So mostly low water area is suitable for cultivation of this plant. And apart from the pharmacological importance, there are a lot of beautifying products. There is aloe vera shampoo, aloe vera skin ointment. If any person who is not consuming aloe vera orally, they will definitely use some products of aloe vera. At least there is a aloe vera gel you can buy from him. There are so many brands, I don't want to quote. The aloe vera soaps are there. So in case of beautifying products, there is great use of aloe vera today. And it contains most of the compounds you can think. There are a lot of anthraquinones, lot of amino acids, then sterols, some hormones, different type of enzymes. It is an house of a source of good vitamins, then saponins, lignins, and some type of sugars that may be monosaccharide, disaccharides. So it is an house of many types of trace elements, minerals. So you can see these are some, it contains almost 75 active ingredients. That might be lignin and some anthraquinones. I have enlisted the trace elements, then anthraquinones, aloin, aloimodine, anthron, and different type of enzymes that may be lipase, peroxidases, sterols, some salicylic acid. Just because of the presence of the salicylic acid, people use on their face as a skin ointment. And then anthraquinones. Basically, these anthraquinones are present in aloe vera in form of aloin, and two isoforms, aloin A and aloin B is there. And these anthraquinones have antibacterial, antifungal properties. And there are around 12 important anthraquinones are present in aloe vera. And the major secondary metabolite is aloin. As we know, the secondary metabolites are produced by plants for their host defense mechanism or to tackle different stresses that may be biotic, abiotic stress may be there. So two forms of aloe vera are present. You can see in the picture, the outer covering, and then below the outer covering, there are the production of the aloin takes place. So most of the aloin that resides in aloe vera just below the outer thick layer. Then it has different properties like antioxidant, anti-aging, anti-ulcer. Again, I'm quoting that you can think which property aloe vera do not have. As the previous researchers quoted that it is have anti-tumor property also. It has an anti-aging property like vitamin A, and it has an, even some researchers claims that it can be used to treat even AIDS. So there are a lot of pharmaceutical importance of aloe vera are there. Then how aloe vera can be used or people using? So it can be consumed orally in form of some juices, capsule form is there, in drinks, in concentrates, in as an flavoring agent, and you can use aloe vera directly on your skin, on your as an hair mask. But the question is, as many researchers reported that aloe vera is too much beneficial, then the question is, it is real. Is it real or not? As it comprises different type of secondary metabolites, active ingredients that are being used as for pharmacological importance, but people are consuming or using aloe vera without knowing their harmful effects. As the previous researchers said that there is production of some secondary metabolites in the desert conditions. So the secondary metabolites are produced by the plants to tackle different stresses, but it is not sure that these secondary metabolites are important. As mentioned by different researchers, that these secondary metabolites can be harmful. If we are consuming unknowingly, they may be potential harmful ingredients. So we have to just check it out whether it is important or not. As Dr. Pankaj said, there are a lot of desert legumes. There are too many legumes which have anti-nutritional components. So we have to identify these anti-nutritional components, or by using different strategies, genetic improvement can be done so that we can minimize these anti-nutritional compounds. So the aloe vera, people consuming unknowingly. So the National Cancer Institute in 1998 nominated that aloe vera is a potent carcinogen. At that time, they do not know what is the exact reason behind this. So this causes genotoxicity, mutagenesis, carcinogen based on the tropical consumption. Then there are some adverse effects these are reported by different scientists, researchers, that is, it is nephrotoxic, then carcinogenicity is there, genotoxic, cytotoxic, purgative effect, blood clotting inhibitor. As many researchers claim that it is a good source, or we can use as an wound healing component. But the later studies suggest that it is a blood clotting inhibitor. So it will be, if you are using for the wound healing purpose, it may be lethal. Then hepatotoxicity. Actually, people are nowadays consuming lot of things just because of the Google doctor. Actually, they just find something, what is the use of that particular plant, and they are consuming unknowingly without knowing their harmful effects. There are several evidences that support aloe vera is not a universal panacea. It has some harmful effects. So in the first study, a 13 days exposure of some rats with the drinking water that is treated with the whole extract of aloe vera. Then there is in goblet cell hyperplasia is reported. There is inflammation in the goblet cell of the intestine. Then the study is conducted for further 2 years. That is, clear-cut evidences are there for the large intestine carcinoma. So it will act as an carcinogen. Then in another study, mouse lymphoma study, that said that whole extract of aloe vera that that is a tumor-inducing compound, contains some tumor-inducing compounds. Then the companies go for the decolorization of aloe vera. As many reports suggest that if we decolorize, then the maximum carcinogenic components will be destroyed. But this is not true. If we consume decolorized aloe vera, it will increase the production of oxygen-free radicals. And these radicals will damage your DNA. So it is not clear that if you will consume decolorized aloe vera, that will help you out. Then again, free radical generation is there. Then genotoxicity caused by aloe vera, it will inhibit the topoisomerase 2, which is an essential enzyme for the recombination of DNA. So it will lead to diminish the activity of topoisomerase 2. Then another study in the root tip of Allium cepa, there is some reports suggest that there is a variation in the chromosomes, or alteration in the chromosomes were reported. Then in the person who is suffering from liver disease and he consumes aloe vera continuously, then the final report suggests that it will lead to affect the alcohol dehydrogenase enzyme. And this is very important liver enzyme. If we will consume alcohol, this will neutralize it. So if we consume aloe vera continuously, it will diminish the activity of alcohol dehydrogenase. So that will affect your liver. Then hepatotoxicity is reported in the labs. Then it causes the aggregation of platelets at the wound site. So it may inhibit the formation of wound exactly. Then it will lead to the male infertility. If we are consuming continuously, then there is a production of enzyme, that is nitrous oxide synthase, and this nitrous oxide synthase will elevate the level of nitrous oxide, and this nitrous oxide that particularly inhibit the synthesis of testosterone production. So it will also related with the male infertility. And the major reason, later researchers found that the major reason of this hepatotoxicity or the adverse effect of aloe vera, just because of the presence of major secondary metabolite, that is aloin. Aloin is metabolized by intestinal bacteria into aloamodine anthrone. And this aloamodine anthrone is a potent carcinogen. So we just have to check then how much aloin is typically drink. There is no clear-cut regulation by any drug regulating authorities or any health department. This is not clear how much amount of aloin is recommended. So actually the industry use 10 ppm, self-regulated industry, they use 10 ppm, but their do not label on the bottle of your juice or your product. So just simply normally think that it contains 10 ppm. But the time is to regulate whether which amount of aloin should be present in the formulations. Then the question is, should I stop drinking aloe vera? You can use aloe vera, you can consume orally, but you should know what is the exact amount of aloin is present in that particular aloe vera. Last night we have had discussion with Professor P.K. Singh. He said that the traditional practices are equally important to the basic research. So we can consume aloe vera. We are consuming from thousands of years. So we can consume, just take a leaf, cut from the bottom, then hang it out. Then there is a release of some yellowish compound, that is sap of aloe vera. Then again cut some part of that leaf. Then will there another drainage of that particular sap is there. So maximum sap that contains the aloin. So if you flush that sap, you can use that aloe vera. But I recommend one thing: remove the cuticle layer. You remove the upper layer, thick layer of aloe vera. Then you can consume aloe vera by orally or by topical applications. You can see there are a lot of street juice vendors nowadays selling aloe vera juice or some different mixture of the leaves of the plants. So first you should know about the toxicity, then you can consume these things. Otherwise it will leads to the health elements or the problems. So how it is regulated? Aloe vera is regulated. It is not regulated because it is not a drug. It is a dietary supplement. So it is not under regulation. Government have to make some policies to regulate the aloin content. Otherwise people consuming unknowingly, and it will show definitely the harmful effects. Thanks. And this is our department, Bio and Nanotechnology, GJU SNT Hisar. And today is the 132nd birth anniversary of Dr. Bhim Rao Ambedkar Baba Sahab, the architect of the Constitution of India. So we are celebrating 132nd birth anniversary. Thanks a lot.
Thank you, Dr. Ravinder, for such a wonderful talk on aloe vera. And usually we consume aloe vera in various products and the various applications of other products. And you have shown the dark side of aloe vera. So it is of, we can say, the factual information. We usually don't care about these things. So the session is open for the small discussion.
And one thing I want to add, by using CRISPR-Cas9 or by some genome editing technology, we can eliminate or we can suppress the expression of the aloin biosynthesis pathway genes. And the study for the reduction of aloin content is going on in our lab at the final stages. So within 6 months, we will try to release or try to publish that research with low aloin content aloe vera. Thank you.
Doctor.
Yeah, you are audible, sir. Yes, sir. Yes. Yes. That should be in, sir. Yes. Yes, sir. Yes, sir. That should be regulated, sir. In front of some parks, you can see there are some vendors who are selling mixture of leaves of different plants, including aloe vera leaves, sir. So they should be regulated.
Dr. Rashid, we have another expert from the Food Safety Authority, Delhi government. So he can tell us better. We have the Dr. Naresh Kumar Sharma. He is currently the Food Safety Officer in government of NCT.
Yes, yes, sir. Yes. Yes, sir. Okay. Actually, FDA recommends below 10 ppm. If you are consuming below 10 ppm aloin, that will help you out. No, US FDA US, sir. They are recommending that if you are consuming less than 10 ppm, it will be beneficial. So the company are just mentioning that we are preparing below 10 ppm aloe vera juice, but there is no recommendation, sir, and they are not labeling on that particular juice bottles. You can find different aloe vera juices from different companies, actually with some amla juices, etc., different type of formulations, but they did not mention that what content of aloe is there. So it should be regulated.
Hello.
Have to be
Sir.
implemented.
Sir, actually, regulations has a secondary role. First role is regarding awareness. As Dr. Ravinder has said, it has anti-nutritional factors or something like that. We are consuming so many things like pulses that have anti-nutritional
Exactly.
factors. But so we should have some research on that. Okay, how we can reduce these type of factors, and regulation is I think it will take much time.
Yeah. Thank you, sir.
Last question, Dr. Ravinder. You mentioned that the aloe vera, it is the good source of vitamin B12. And you will be surprised to know that in India, there is 46% of Indian population that is suffering from vitamin B12 deficiency. So is there any work going on in this direction in India specifically?
Actually, you can, there may be some strategy or the future recommendation that you can isolate vitamin B12 from these and then you can consume. If you will consume directly, as I said earlier.
Can we take the help of that CRISPR technology for this?
Yes, definitely. And one more thing I want to add. People are consuming or companies are just procuring aloe vera from Rajasthan because it is a zero phytic plant, require less water. In the stress condition, the secondary metabolite will elevate maximum folds, many folds. So if you are consuming aloe vera that is particularly from Rajasthan and you are consuming lot of aloe, and that will help to the different diseases, lead to different diseases.
Yes, thank you.
Thank you very much.
Please.
There is no particular technology developed that we can just remove the aloe from the aloe vera juice. You can only cut that leaves and hanging it out, then sap will drain out. But it is not for sure that how much amount of aloe still the aloe vera juice contain.
Yes, the study, actually, I joined the session late, so I had not gone through all the slides. So you mentioned the anti-nutrient, toxicity of
Yes,
aloe. So is there a study available with the dose dependent study?
The rats were treated with different type of doses. Actually, the dose dependent, so that we can see that
in that study they had mentioned that the amount, how much amount is not toxic to the,
as I said, below 10 ppm.
Below 10 ppm.
That is recommended dose. Okay. If you will consume below 10 ppm, that will help you out. If you consume more than that, it may lead to some disease.
Okay.
Thank you. Thank you.
Sir, I would like to say that, as you said, so we need to work on the processing techniques. So there are many anti-nutritional factor in many foodstuffs. But when we are using some processing techniques, like for phytates, we are using like roasting, germination and all. So such kind of techniques we also can develop to reduce the aloe content among the aloe vera.
Yes,
so that can be one of the point of the study. So further
Exactly.
we can do in study.
If we can develop any bioprocess technology
Yeah.
by which we can remove alloin for the during juice
And
preparation.
definitely we can remove, because we have to explore this area.
Actually,
also.
major problem is that alloin directly is not carcinogenic. If we consume aloe vera, then intestinal bacteria that will convert alloin into aloimodine or aloimodine anthrone. These are the potent carcinogens. So you can remove these alloins in the bioprocess technology by using bioprocess technology. That will help you out.
Yes.
So there is need to develop some bioprocess technologies.
Techniques, yes.
Yes. Thank you.
Thank you, Dr. Ravinder. And the discussion shows that how much the topic was interested, and based on the popularity of the aloe vera. And thank you, Dr. Ravinder Kumar, for this wonderful talk. And we have the next speaker, Dr. Neeraj Srivastava. Dr. Srivastava is Associate Professor of Veterinary Microbiology in Veterinary College, Rewa, Madhya Pradesh. Dr. Srivastava will speak on different methods for the study of bacterial biofilms. Welcome, Dr. Srivastava.
So good morning, everyone. First of all, I would like to thank the team MET and particularly Dr. Ashok for providing me the opportunity to deliver a talk on the subject of different methods for the study of bacterial films. See, biofilm is defined as a sessile microbial community in which bacterial cells are attached to a surface and embedded in a self-synthesized protective extracellular matrix. Actually, here, this is a microbial community. It consists of many microbial organisms, many types of bacterial species, and the matrix is synthesized by the population itself. Actually, biofilm is a major concern for the public health, for in the medicine and in the food industry, because it is causes problem in the treatment, and also it damages the food equipments. The biofilm is because the antibiotics cannot attain the particular concentration when we are infected with a biofilm positive organism, and it causes resistance to antibiotics. And in the food industry, this biofilm is related to the public health as well as for the food safety. So the biofilm is a matter of very intensive research. And to understand these methods, we will first see there are three main steps of in biofilm development. And first is that the microorganisms will attach to a surface. It could be a biotic or a abiotic surface. And then which is a very tightly regulated process, they will form the film, they will secrete the matrix, and then they attain a 3D structure with fluid channels. And then the same microorganisms, they will change their metabolic profile, and then they will change and disperse, and then that will again form biofilm at a new surface or a new area. So in this, these are the main areas. So basically it is all related to the like the research is concentrated on whether we can prevent at all the formation, or when it is already formed, then we can disperse it. So as you can see, the microbial composition, this biofilm composition is that the cells are only 2 to 5, DNA is less than 1, polysaccharides 1 to 2, proteins are also all in the range of 1 to 2, and it's more water. So here, when we talk about in methods, when we are using the scanning, then we have to take care of the dehydration step. And to make the things more complicated, it is just to show you that it is a multi-species community, and it is regulated, and the components are also very different. So it makes, the study a bit more complicated. Traditionally, the biofilm is studied as a direct and indirect methods. But when we started studying it, I thought that, this was more useful way of planning the experiment, because the access to the instruments made our experimental design, were influencing our experimental design. So I have classified it into spectrophotometer based or fluorometer based and the microscopy based methods. So the most common one is the crystal violet method, and the most common microscope which is used is scanning, confocal laser scanning microscope. So here you can see there are, the measurements are of adhesion, cell viability, cell biomass, metabolic activity, biomass, biofilm mass, and matrix composition. These are the main things which we can study in biofilm. And these are the instruments which we will use, or the techniques. Mainly the production is done by the continuous flow or the static, or there are certain very microfluidic models. But the most commonly used is the 96 well titer plate. So the spectrophotometer based is this is the technique where we grow the bacteria in certain biofilm enhancing compound, and then we stain that, and then we de-stain it, and then find out how much crystal violet is there, which is directly related to the biofilm mass. And there are a lot of influencing or variables which make it a very challenging for comparison of one study to the other. And this is the way, and this is of Staphylococcus aureus, in which we work, and this is the way it is interpreted. How to be classify it as a weak or a moderate or a strong biofilm producer. Although it is very regulated, but this is the phenotypic expression. This is when we want to, because in crystal violet we are killing the cells, so everything is sustained. And when we want to see the metabolic activity of the organisms in the biofilm, then we use tetrazolium dye-based methods. And the most commonly one is used is risazorin, which requires spectrophotometer. Then the microscope-based methods is the confocal laser scanning microscope is the most common way. And the most sophisticated one is atomic force, which we can use to define the strength of the biofilm. So this scanning microscope, and we can visualize the 3D structure because it is minimal invasive, and the spatio-temporal structure also, the composition, and we can even identify and we can even study the formation of biofilm in real time. So live and dead staining to see whether our product is killing or not. So this is the most common ones. This for the staining of the extracellular DNA or the lipids or the carbohydrates or the proteins. And when we are using it, and then we require image analysis software. So the ComStat is the most freely available, and that is specific for biofilm images. And Imaris is a proprietary, while others are free to download. And this is, it can provide you the fractal dimensions of our biofilm, which we are studying. And as you know, this is a typical problem with biofilm, that the variation is very inherent in the design itself. So it is usually done in triplicates, and 3 times that same experiment is repeated in different days to minimize it. And this is the initiative which provides the certain standards and guidelines when we are planning the experimental design, so that the variation is minimized and we can compare the one study with the other. So to conclude, the biofilms are 3D structures of multiple organisms which provide protection against the antibiotics and disinfectants. And the research is based basically to prevent and or to disrupt the already formed biofilm. The microtiter based method is the most common and simple method, where you don't require much of facility and is very easy. Confocal is the most widely used microscopic method to study the 3D structure and time and space, but it is very skill intensive also. So, thank you.
Thank you, Dr. Srivastava. The session is open for small discussion. Okay, thank you, Dr. Srivastava, for your talk on the biofilms. Now we will have a 15 minutes break for the tea. So we will join the tea, and after 15-20 minutes, we will again start the session. Thank you.
It is Dr. Astha Diwan. Okay, so our next presenter is Dr. Nikhil. So you have 7 minutes for the presentation.
Hello. Start में. Okay. Good morning to all of you.
Yes, sir. वीडियो स्टार्ट कर लिया
Very good morning to all of you. My presentation topic is evaluation of public distribution system in Himachal Pradesh. So, as the introduction part is concerned, we know that basically public distribution system is the system in which government provide essential commodities to the weaker section of the society. And this scheme were introduced in Second World War during the British government. It is basically a food security system of India. And currently more than 5,47,000 of fair price outlets are prevailing in India. This is the outline of public distribution system. First of all, in 1940, this scheme was launched at general entitlements. And after that, after a very long time, there is a remap in this scheme. And in 1997, a special focus, that is target public distribution system, was introduced, in which especially the uh, weaker section of the society or poorest of the poor targeted under this scheme, that is also known as Antyodaya Anna Yojana. And recently in 2013, September 2013, a act that is known as National Food Security Act, or this act is also known as Right to Food Act 2013, were introduced in order to for the food security purpose. And this act are concerned, it has three components. First component deals with public distribution system. Second component deals with midday meal scheme. And the third component deals with the integrated child development services scheme. So my basic focus on the first part of the this component, that is public distribution system. Share.
ये बार बार
So this is the network of public distribution system. Basically, the whole system is divided into three categories or three types. First one is the procurement, second one is the management, and third one is the distribution part are concerned. As the procurement part is concerned, for the for procure the foods, National Food Corporation of India has been established with its headquarters at New Delhi, and it was established basically to the food grains trade. And the second part is management part are concerned. In management, there are various agencies, like for example, at the central level, Ministry of Consumer Affairs under the Department of Food and Public Distribution System are there. Second one is the at the state level, Department of Food is also there. Civil Supply Corporation is also there, and State Civil Supply Corporation are also there. As the distribution part are concerned, there are various agencies who distribute these essential food grains to the peoples. So these distribution agencies also include the fair price outlets. It also run by individual. In some state, it is run by the state civil supply corporations, self-help group, and so on. This is the working mechanism of the PDS system. There are basically four pillars of this system. First is the farmer or producer. Second one is the Food Corporation of India, where buffer stocks are maintained. Third one is the distribution outlet, that is fair price shops, and it also includes the respective government also. The position of public distribution system in Himachal Pradesh currently, it have more than 5,000 outlets, which are run by, for example, 3,245 are run by cooperative sector, that is Himachal Pradesh State Civil Supply Corporation, 1,656 by individual, 69 by Himachal Pradesh Civil Supply Corporation, 18 by Mahila Mandal, and 12 by the Panchayati, and 1 by self-help group or individual are concerned. This is the literature review part of our concern. From time to time, there are various researcher or author, for example, Mahindra and Indrakant in 2014. Basically, the literature review focus on the awareness, problem part, and satisfaction level of the beneficiaries. As the objective of the study, there are three objectives of the present study because the study is descriptive in nature. The first objective deal with to study the awareness level of the beneficiary regarding public distribution system. Second one is to study the satisfaction level of beneficiary regarding public distribution system. Third one is to find out the various problems by beneficiary or respondents regarding public distribution system. As the need of study are concerned, because the study area is Himachal Pradesh, so we know that Himachal is a hilly state and living conditions are quite difficult. Agriculture is the mainstay of the people of Himachal Pradesh, and more than 90% of the population relied upon agriculture and its allied activity. Now in this hilly and mountainous state, just like Himachal Pradesh, where according to the Department of Statistics and Economics, almost 10% of the population living below poverty line. So it's become the respective state government duty to provide necessary food grains to the respective respondents or beneficiaries. As the hypothesis part are concerned, based on these objectives, because the objective are based on the research question, these hypotheses have been framed. Null hypothesis: there is no significant difference in the awareness level of respondents toward public distribution system. Alternate hypothesis: there is a significant difference between the awareness level of respondents towards the public distribution system. Null hypothesis second: there is no significant difference in satisfaction level of respondents towards public distribution system. And null hypothesis second: there is a significant difference in satisfaction level of respondents towards public distribution system. As the sampling design are concerned, because the study is descriptive in nature, multi-stage sampling has been introduced. In the first stage, there are 12 administrative districts in Himachal Pradesh. Out of 12 districts, 4 districts has been chosen in descending order based on the proportionally sampling basis. These districts are Kangra, Chamba, Mandi, and Shimla respectively. In the second stage deals with the selection of the blocks are concerned. So from each district, 3 blocks have been selected, and there are 12 blocks from 4 districts. In third stage is related with the local self-government, that is Panchayati Raj institutions, and from so this is the conclusion part. So we know that the public distribution system caters to the need of the rural people, especially the weaker section of the society. And from the entire study, the sample were 550 respondents. It is found that the respondents are facing many types of problems regarding this system. Fair price shops and adulteration is the main problem faced by the beneficiaries, and somewhere dealer are compel beneficiary for the unwanted goods to purchase the unwanted goods. And providing food grains to the people is an important part of food security policy of the government. There is also a need to improve the system at various levels. Thanks. Ma'am, because there are three objectives in my study. First one is to check the awareness level of the beneficiary. Second one is to know the satisfaction level, and third one is the problem faced by different beneficiary. To accomplish this objective, the statistic tool arithmetical mean and standard deviation has been used. Because the question is well structured on 5-point linked basis. As the awareness part is concerned, it is found that consumer are pretty aware or moderately aware, but not fully aware, because the sample size is from Himachal Pradesh, and rural people are not so much aware and not so much know the objectives of these government schemes. Sir, there are some problem like, for example, adulteration is the main problem or concern, and there is a lack of at people level. There is not properly displaying the menu list outside the PDS outlets. There is also a problems consumer or beneficiary or respondents not getting their food items on a time basis, and so on. Okay, thank you.
So I would like to call Dr. Priyanka Pare. Yeah, good afternoon, everyone. As we all know that food sustainability is one of the important aspects nowadays, and it is very important to get the nutrition security. And nutrition security is very important to get the sustainable nutritional status. So that's why it is very important to focus on the sustainability of nutritional status. So in our institute, earlier we have conducted the studies to assess the awareness or the knowledge among the patients. I am from the clinical nutrition departments. So basically we focus on the patients. So earlier we have conducted the study to assess the knowledge and awareness among the patients. So in the same line, we have assessed the knowledge and awareness among the chronic kidney disease patients. And then we got to know that they are not so much aware about the nutrition. They are not so much aware about the foods to be avoided and foods to be included in their diet because they are on dialysis. So it is very important for them to know about the about the proper dietary guidelines. So that's why in the same line, we have planned and we have conducted the study. The title of our study is Impact of Nutrition Education Intervention on the Nutritional Status of Hemodialysis Patient in a Tertiary Care Hospital. Sir, ये down नहीं है. यहाँ से होगा.
नहीं PPT.
So the title of my study is Impact of Nutrition Education Intervention on the Nutritional Status of Hemodialysis Patients in a Tertiary Care Hospital. So as I said, it is very important to maintain the nutritional status of the patient. And as we know that prevalence of chronic kidney diseases are increasing day by day in India also, and it is almost 17%. And increase, so in these chronic kidney disease patients, there is a chances to develop malnutrition. And if the malnutrition is there among the hemodialysis, the patient, those who are on maintenance hemodialysis, then day by day, their health will be deteriorate and the death chances will be more and the hospitalization chances will be more. So that's why it is very important to maintain their nutritional status. So to maintain their nutritional status, it is very important to plan some nutrition education intervention for the patient and for the relatives so that they will be able to maintain their food intake, their dietary intake, and to avoid the malnutrition among them. And as we all know that it is very important to take care about their protein, and then it is very important to take care about their electrolyte intake also. So that's why I have planned the study. The objective of my study is to find out the impact of nutrition education intervention on the nutritional status of hemodialysis patients in a tertiary care hospital. The patient, those who are on maintenance hemodialysis. So this is the methodology of my study. It was interventional study conducted in the Department of Medicine, Dialysis Unit, MGM Hospital, Kamothe, which is situated in Navi Mumbai. It's a tertiary care hospital. So we have taken 70 dialysis patients on the basis of following inclusion and exclusion criteria. The patients more than belongs to the age group of more than 18 years of age, and patients, those who are regularly visiting the hospital for the hemodialysis, and those patients, those who are willing to participate in the study. And the patients who are suffering from any other chronic disease like cancer, HIV, AIDS, and liver diseases, we have excluded them. So we have selected 70 patients on the basis of that. And then randomly, we have divided the patients into two groups. One is the control group and one is the experimental group. So each group contains 35 patients. Okay, and after that, for the further investigation, first of all, we have taken the ethical approval from the institutional ethical committee, and then we have taken the consent from the patients also. And after that, this is a part of pre-investigation, pre-intervention investigation. Yes? No, but it was glitch between them. Yeah, that is okay. That is okay. So socio-demographic profile, height, weight, BMI, body composition, and dietary assessment we have assessed in biochemical parameters also. Then we have given them the nutrition education intervention for 3 months. And then again, post intervention, we have conducted the same investigation. Then we have done the data analysis by the SPSS software, and then we have drawn the results. So these are the socio-demographic profile in that you can see the males were more as compared to the females. 43% were in the age group of 40 to 60, and some of the maximum were having the non-vegetarian food habits. Here you can see the changes. Here I have presented the changes pre-intervention and post-intervention. In this, you can see that there are the significant changes you can observe in the experimental group pre- and post-intervention. So their body fat also, visceral fat also increased, their body weight also increased. We have seen the significant changes in their muscle mass also. So in the intervention, we have given them the knowledge about the foods to be avoided, foods to be included. We have planned the multi-component nutrition education intervention for the patient. For 3 months, we have taken their follow-up also. And here I am presenting the results of the same after the intervention. So we have seen the significant changes in the experimental group. So this is the body composition. Here you can see the shifting of the patient from the undernutrition to the normal category among the experimental group, because we have given them the nutrition education, we have given them the diet chart also, we have forwarded them the videos, audio visual aids also, and then the printed material also. And then you can see the same changes in their biochemical parameters also. These are the biochemical parameters which is important for the chronic kidney disease patients. Here you can see the significant changes within the group also. Within the group means the experimental group before the intervention and after the intervention. So significant changes in the albumin level, protein level, globulin level, hemoglobin level, and the calcium levels also. And then here we have taken their dietary recall also by the 24-hour recall and food frequency. And here you can see the changes in their nutrient intake also. So there is a significant improvement in their energy intake among the experimental group. In the significant changes we have seen in the protein intake, carbohydrate intake also we have seen, and in the calcium intake also because of the intervention. And these significant changes we have observed in the experimental group after the intervention. So overall, one more thing we have observed, because we have included the foods to be avoided and foods to be included, the lower potassium containing food items and all now. So we have observed the changes in their food choices among the experimental group. So now they have started to collect the protein-rich foodstuff, potassium-containing foodstuff, and they have cut short their fluoride intake also. So that is the positive side. So on the basis of that, I can conclude on the basis of the results from the study, we can see the positive effects of nutritional education on the overall nutritional status and different dimensions of the health among the hemodialysis patients. So the study can be summarized by saying that the comprehensive dietary educational intervention must be incorporated as a specific approach to facilitate patients' health status and reduce the burden of complications among the dialysis patients. So I can conclude that if we want to get the sustainable nutritional status among the patients, then also nutrition education intervention is one of the powerful strategies. Because every time we are giving weightage to the food approach, we are giving weightage to the supplements. But if we are not giving them the education, then it's of no use, because the people are not aware about that. Even they are going for the maintenance dialysis, but they are not aware what kind of foods they need to take, what kind of foods they need to avoid. So this can be one of the sustainable strategies to combat the malnutrition among the disease patient, not for the chronic kidney disease patient. For any kind of chronic disease, this can be the powerful strategies. And even in our institute, we are working on that. Thank you so much. Yeah, the thing is, this was a pilot study, and in our institute, at a one time, we are registering 80 patients for the dialysis. We have the dialysis unit. So among the 80 patients, only 70 patients were eligible for my study. So that's why, and they have given the consent also. So that's why I have taken. Further, we are planning a multi-centric study also, because in our we have lot many other hospitals also, which is affiliated to MGM Institute of Healthcare. So definitely we are planning. We have other dialysis units also. So this was the pilot study. We are planning. We are in a process to plan the intervention study for that. No. We are, it's our protocol. So we are taking the 24-hour dietary recall for 3 days. Yeah, that is in our process. And even we have taken the food frequency also. So that is also one of the important aspects. So because of the shortage of time, I was not able to explain you about that, but it's a protocol. So we are taking. Education, yes, sir. Sir, we have used the multi-component comprehensive approach for this thing. So we have given them the one-to-one counseling also. That is a patient customized diet also we have given them. We have prepared the audio visual aids also by the help of the dietitian who is working in the nephrology department. So content validation also we have done before the execution of the intervention. We have planned a printed material also for our patients. We have handed over the printed material to our patients, and we have taken the follow-up of our students within this 3-month duration by the mobile phone also. We used to give them the messages also. Yes, as a reminder, yes, you need to take this, avoid this, take this. So such kind of all these components we have utilized for our intervention. Yeah, thank you. Yeah. That's why I explained and I told na we have used a multi-component comprehensive nutritional strategies. So we have moved step by step. One-to-one counseling is also very important. In the first meeting, we cannot expect or we cannot explain the patient so much in detail about the diet. First of all, I should know the attitude of the patient. So that is very important. So it's a step-by-step process. Even we can plan for 6 months also. It can be a long-term study also. So we are in a process to plan a long-term study because long-term study will give better result as compared to my study. Yeah. Thank you so much. Yes, sir. Sir, we are using body composition analyzer for this bioelectric. This body composition analyzer works on the principle of bioelectrical impedance. So this we are using for the patients. Sir, the apparatus itself gives the readings for different kinds of parameter like muscle mass, visceral fat, body fat. All these things the apparatus will give because it works on the principle of bioelectrical impedance. So the electricity passes throughout the body. So fat, it gives an account of fat also. It gives an account of muscle mass also.
Does it differentiates the subcut fat and internal fat and this visceral fat?
Yes.
There are different types of fat in body.
Yeah. It gives the reading about all kinds of fat. So there are different types of body composition analyzer. You can go towards the most sophisticated or most advanced also. So there are the different ranges of such kind of analyzers.
Okay, thank you.
Thank you.
Thank you, Dr. Priyanka. I would like to call Mr. Shifali Tripathi. And there's a reminder, please adhere to the timings. Please.
A very good afternoon.
A very good afternoon to everyone. My name is Shefali Tripathi, and I'm here going to present my research topic, that is UV blocking antioxidant properties of chitosan-based packaging material infused with acacia catechu and calcium carbonate for the food safety. As we know that food safety and security has been a major issue, and around 1/3 to 1/4 of the food get wasted without the utilization. And as we all know that there are many people who are dying due to this food scarcity because of the expired shelf life and oversupply, or not having the proper flavor or textural differences. So food packaging will help active food packaging will help to preserve the food and increase. Okay. Okay, but like it was like just the basic. So the oxidation of the food is a major issue, and it leads to the spoilage of the food and deteriorate the quality and hence decrease the shelf life of the food. So here we have utilized this acacia catechu, which is also known as pan katha. So this has the properties which depicts the antioxidant properties, and it also has it act as a coloring agent, and which can be utilized for the light sensitive food products. So basically here in the systematic overview, we have firstly extracted this acacia catechu, and we have combined it with the chitosan based film in combination with the calcium carbonate, and then we have developed these films, and further we have done this characterization. These are my results. These are the physical properties, that is thickness and color parameters. So as you can see in this in the film we have developed, as the concentration of the acacia catechu has increased, the opacity or the color of the film is enhancing. So these that's why these films can act as a barrier to the UV lights. Then we have done several other tests for the packaging material, that is water vapor permeability, tensile strength, elongation rate, moisture content, and water solubility. This is the UV transmittance test we have done, and it was observed that the highest concentration of the film, that is CHCCCT4, has the lowest transmittance of 0.46%, which depicts that yes, these can be used in the packaging material. Further, we have done this antioxidant test, and for this we have done this DPPH radical scavenging activity. And as the concentration of this acacia catechu we have increased, the antioxidant properties of the film has also increased. Then this is to confirm the antioxidant activity. We have further done the PRAP test also, and similar results according to the antioxidant were found. For the surface morphology of the film, we have analyzed this FESM and compared it with the neat chitosan-based film. Further, we have done to analyze the functional bonding between the compounds. We have done this FTIR test and find out what is the significant difference we have analyzed while adding the acacia catechu in the chitosan-based packaging film. For the thermal stability of the film, we have done this TJ analysis, and it was observed in the 3 phases. That was firstly between 100 to 200, and further it was 200 to 500, and after that 500 to 700. For the crystallinity of the film and how it can impact the tensile strength, we have done this XRD test, and from this we have depicted that it has a semi-crystalline nature. So the conclusion is that these packaging film has the ability to be utilized in the for the packaging of the material which are light sensitive and which has high lipid content, as they are more prone to oxidation. So better the packaging, safer is the food. Thank you. And these are my lab members. So the question is welcome. Sir, we haven't analyzed the sensory attributes as we are not focused on the edible part. Like we have not claimed that it is an edible packaging film material, but we can. Uh, yes, sir. Yes, sir. So we can do it in the future for sure. Like this. So we have it will act as a inorganic filler, and it has enhanced the tensile properties of the film. Thank you, ma'am. Thank you.
Thank you, Shefali. So I would like to invite Praveen Kumar.
Good afternoon, everyone. Today, I am going to present the topic of effect of hollow-site nanotubes and microfibrillated cellulose incorporation into the pine needle paper to use it as a ethylene scavenging paper. Basically, I am Praveen Kumar Ponam from IIT Roorkee, and currently I am working under the professor of Kirtari K. Gaikwad. This is my contents. First, I will go through these all contents, and next, coming to introduction, there are so many disadvantages associated with plastic. We all know. For best, we can replace the plastic packaging materials with the paper-based alternatives, which are shown here. And if you can see, annual food loss, actually 1.3 billion tons of food is getting wasted each year. And one of the main reasons for this food loss is ethylene absorption. If you can see normal packaging, generally these kind of fruits, climatic fruits, will release ethylene, and that will fasten the ripening of fruits, and it will spoil the foods in faster manner. And active packaging is a manner which will absorb these ethylene, oxygen, this kind of things, and it will enhance the shelf life of the foods. If we can absorb the ethylene and if we can increase the shelf life, we can reduce the food loss. This is our main objective here. And here we are using the pine needle waste. Actually, in annual 4,000 hectares of Uttarakhand land is harvesting pine needle, and it is nearly 1.3 million tons. And it is getting wasted each year, and it is causing forest fires. And we already discussed about food waste. It is also main problem in the current society. And one more thing is plastic. Plastic complete ban on single-user plastic is imposed from July 1st in 2022 from India. And UN also telling food waste should be reduced, and waste to wealth policy, and G20 is telling that we have to be sustainable. By adhering to these all things, we did one thing, that waste to wealth. We utilized pine needle waste to produce a paper which can be used in packaging, and that packaging also in such a way that we incorporated HNT and microfibrillated cellulose, so it will act as ethylene scavenging paper. This is the overview of our research work. First, we took pine needles and we did sun dry. Then we produced the pulp suspension, and at the same time, we used pine cones to produce microfibrillated cellulose. By incorporating this microfibrillated cellulose into the pine needle, at the same time, we incorporated halosite nanotubes, and we produced the active paper. By using this active paper, we can reduce the ethylene. When we pack this kind of fruits, bananas and apples, this kind of fruits with this paper, we can reduce the ethylene quantity, and we can increase the shelf life of the food. This is the process I followed to produce the paper. First of all, we took the dried and cut pine needles. Then we did digestion. Then pulp. We obtained pulp, and then we did beating. Then we added HNT suspension to the pulp suspension. Then we did paper making. Then pressing. Then finally we obtained the paper sheet, which is shown here. Results and discussions. These are the papers which were obtained after the producing. First one need PL. Just it is without any addition of microfibrillated cellulose and MFC. microfibrillated cellulose and HNT. Second one is pine needle paper, which was obtained by the addition of microfibrillated cellulose and HNT. From the FTR studies, we showed that OH groups are present in this paper. That means cellulose and microfibrillated cellulose are present, and SiO and Si. And we know that HNT formula is alumina silicate. That Al and OH peak at 910 and SiO Si peak at 1028 are showing that HNT is present in the given paper. These are the physical studies, strength papers, strength of the paper. Generally, when we are using this paper for packaging applications, it requires strength. So we obtained all the strength properties, and they are showing that microfibrillated cellulose enhances the strength properties of the paper. This is the surface morphology of the paper that we are obtained, and it is showing that HNT is present in the morphology of the paper. And water vapor studies, and the main study is here, ethylene scavenging study. If you can see, first NEAT PL is just reducing small amount of ethylene by the end of 23rd day. And if you can see 20% ethylene, 20% HNT loading is showing 68% of reduction in ethylene, and 30% HNT loading is showing highest, nearly 80% reduction in ethylene. So if we can use this paper for packaging of the fruits and vegetables which can produce ethylene, we can reduce the food waste and we can increase the shelf life of the foods. These are the TEM images which are showing ethylene, that HNT incorporation. My conclusions are, we successfully utilized the pine needle waste to produce a paper, and we incorporated MFC and HNT. MFC increased the mechanical properties and HNT increased the ethylene scavenging properties. Finally, we produced a sustainable packaging material to increase the shelf life of climatic fruits. So we can reduce the food waste by using my study. These are the references we followed to do our research work, and these are our laboratory members. Thank you for giving me this opportunity. No, ma'am. Currently, we didn't apply it to not directly fruits. We just obtained the results that 2030 it is reducing 80% of the ethylene. We just obtained the study. Further, we will do that study also by applying directly to fruits and veg. Stretchability, according to, if you can see, the paper don't have that much stretchability as the polythene papers, but it have good strength properties. It can hold much more amount of food compared to the polythene papers. Polythene will stretch, but paper don't stretch, but it have good properties, strength properties. Thank you.
Thank you, Praveen. So I would like to call Anushika for a presentation.
Thank you. Good afternoon, everyone. I'm Anushika from IIT Roorkee, pursuing Masters in Packaging Technology. I'm working under Professor Kirtiraj K. Gaikwad. Today, I'm presenting my research work titled Gargum-based ethylene scavenging and UV blocking biocomposite film integrated with lignocellulosic nanofibrils and MOF for food packaging applications. So, as we all know that plastic is getting produced each year, and it is the main reason of the polluting the. Oh, okay, yes, sir. Yes. So ethylene is the ethylene works as a hormone which helps in ripening the fruits and vegetables. So, but over excess of ethylene will reducing the shelf life. So, and this all because of the packaging, that plastic packaging. So we can use the ethylene scavenger for reducing the ripening process, and increasing the shelf life. They are in market chemical oriented. Some ethylene scavengers are there, but they are chemical oriented and harmful for health. So, so we have produced ethylene scavenging film, packaging film. I took sugarcane bagasse and sugarcane bagasse for producing lignocellulosic nanofibrils, and I synthesized the MOF, metal organic framework, which is the rich source of ethylene scavenging. So this is my methodology, filmmaking process. I took sugarcane bagasse, then I dried that and I grinded, and then we performed alkali pretreatment, soda pulping, soda bleaching, soft bleaching, and then we got this lignocellulosic nanofibrils, and we have synthesized that metal organic framework with the zinc nitrate hexahydrate and methyl imidazole and plus zeolite. So and plus gargum as a thickening agent we took in producing our film, and these prepared film has the antioxidant, UV protection, and ethylene absorption properties. So these are physical properties of my film. They are as increasing the MOF content, that the color is darkening, color is getting dark. So and the thickness of the film is increasing increasing the MOF content. So and these are the some water contact angle is also increasing, means and increasing as the increasing the content of MOF, water contact angle is also increasing. And these are the SAM images. So we can see here with the increasing MOF content, the some small clusters are there in the surface, on the surface of that those films. And with the FTIR, we can understand here that all groups are present, and the bonds are present here. What are present in compounds? All we have taken. And with the thermogravimetric analysis, we can see that thermal stability is increasing with the increasing the MOF content in the films. In the UV with transmittance, we can see the UV activity of that sunlight. UV activity is decreasing with the increasing the MOF content of the film in the film. DPPH scavenging activity is showing that it is having antioxidant property. Around 40%, more than 40% antioxidant property is there in film. And this is the result of ethylene scavenging. So with the increasing the MOF content, it is getting decreases by the 6th day. It is decreasing. We can see here in the results. So these are the visual they performed on the banana study. Is there we can see we took MOF with a high concentration of MOF film, and we took one control film which is gargum based. So we can see here that till the 8th day, that control film that covered that banana got spoiled totally, and in the MOF content that film, it is showing it is little black points are there. So we can say that it is increasing shelf life and decreasing the ripening process as so fast. So these are my conclusion. These films having antioxidant, UV property, and ethylene scavenging properties. Well, so these are references, and this is my lab members who are working in food packaging. Thank you. Six samples control film wala, and 6 samples that yes MOF control.
How this clean scavenging activity was assessed in this work?
So actually,
what was the model you have adopted or procedure?
That we have made those films. Then we have packed in polythene, And those films, one MOF जो है MOF film, we put in those airtight zip bag, and we put those banana. Then we were just testing each day after each 2 days, and we are testing that that it's ethylene is convincing. And sir, ethylene gas analyzer is also there. We have performed that, and we saw that ethylene convincing is getting reduced every day.
Okay. Thank you.
Thank you, sir. Yes, sir. Yeah.
Thank you, Anushika. I would like to invite Prachi Jain for her presentation. Good morning, sir. Good morning, ma'am. Good morning to all the respected professionals here. My name is Prachi Jain, and I am from Indian Institute of Technology, Roorkee. I am a PhD scholar, and the topic of my presentation is novel temperature-sensitive label based on thermochromic ink for hot food packaging and serving application. So basically, before explaining my results and work, I would like to give a brief description on the problem statement which led us to the development of thermochromic ink. So basically, I'm sure that most of us, not if frequently, at least once in a lifetime, or let's say once in a week, we all make use of food delivering apps like Zomato and Swiggy. And in India, it is quite popular. But the basic problem with these so-called convenient food delivering system is that the customer is totally unaware of the state of food. And when I say state of the food, I mean that that as a customer, you cannot know or you cannot assess what is the temperature of the food. When I say this, I want you to think about some very common basic questions like how do you know if the temperature of food was actually hot? How did you know they actually did not delivered you stale food? Did they actually warmed up your food to the required temperature so that you can enjoy your food which you paid off? So basically, to answer these simple but practical questions, we developed thermochromic ink. Thermochromic ink is used in the application of hot beverages, ready-to-eat food, doorstep delivered food and retort pouches. As you can see in the animation also, a basic principle is this: when hot liquid is poured, the label will change its color. So in our study, we use STIM, which is silver tetraidomercurate, and the color changed from yellow to red. The STIM basic color or the original color is yellow. It changes its color from yellow to red, and the temperature zone, to be exact, was 39.8 degrees Celsius, and it will remain red till 59.8 degrees Celsius. This kind of reaction is reversible. So when your food will become cold or when the temperature will drop, the color will again come to yellow color. As you can also see in the picture, at 39.8 degrees Celsius, it is yellow. But when it reaches around 60 degrees Celsius, the color changes to red. This is the methodology we used. First, we prepared the pigment using silver nitrate and Nestle's reagent. And then because we wanted it to apply on practical packaging applications, printing applications, we developed it into coating system. And this is our results. So we did FESEM, that is field emission scanning electron microscopy. The first one, which is A, is actually cellulose acetate layer, which we used as a binding material for the ink and the paper substrate. As you can see, the paper, as you can see, there is a neat film which shows that the binding properties of cellulose acetate is quite well. And with the increasing concentration of STIM, we added it into different concentrations like 0, 50, 60, 70, and 80. And you can see the particle numbers are also increasing, since it is obvious because we are also increasing the concentrations. Then we did colorimetric analysis to see the temperature range. We did it for all the concentrations, but I have shown only for 80. And you can see the L value, A and B value. The A value depicts redness of the ink since our final color was red. So with increasing temperature, the A value is most at 60 degrees Celsius. We also did thermal analysis, and in thermal analysis, we found that that for the developed zone, it is completely safe. It will not degrade. We did rub resistances. So basically, rub resistances was quite important since we wanted it to apply in practical packaging application. When a coated label is actually printed on a coated box, it encounters a lot of abrasive forces. Even in printing itself, there are a lot of rubbing and tearing which happens and which can scab off the printing. So we did rub resistance test. The after shows the scubbed area, which you can see, as you can see, there is no significant changes after rubbing 20 cycle tests, which means the label was quite adhesive to the surface and it did not turn off. We also did thermochromism of coated label, which means like in neat CA, there is no change because we did not added any STIM. Then 50, 60, 70, 80, and the best results were obtained for STIM 80 because the yellow color changed to red at 333 Kelvin, which is actually 60 degrees Celsius and now. So for conclusion, I would like to say the developed novel color changing system based on temperature is kind of an advanced packaging system that communicates the state of the food, and it mainly focuses on consumer convenience. Thermochromic ink can not only communicate about the food's temperature, but it may also communicate about the freshness of food. We also did adhesion test and solid content test to understand the coating, and it can be said that the coating can be developed for printing ink, which can even be more convenient for food packaging applications. Thank you. Ma'am, actually, STIM, we are not making it in direct contact with food. We are rather sticking it on the packaging itself. And when printing of ink takes place, there is a very thin laminate of plastic behind both the sides of the printing, so it cannot migrate into the box. It is only restrained to the inking part also, the laminate film part only. So it is not coming into the direct contact of food. Yes, ma'am. Thank you. Thank you.
Thank you, Prachi. I would like to call Isha Gupta. Yeah. I don't know.
Good morning, good afternoon, judges and all the respected delegates. My name is Isha Gupta. I'm a PhD scholar in Department of Biochemistry at CSIR CFTRI. I'm very grateful for being able to present my work here in AMIFOST 2023. I'm basically working on nutraceuticals effects on metabolic in relation with metabolic disorders. And for today, my topic is, how can I remember this? Yeah, the topic is anti-obesity effects of oleoresin extracted from Nigella sativa seeds on serum lipid profile, gene expression, and in situ tissue morphology in high-fat diet-fed C57 BL6 mice.
Okay. Obesity, as we all know, is a worldwide epidemic which we as a population are dealing with. Especially in India, there is a hike in cases in the last 2 decades, especially in the adolescents and young age adults. So there are many causes of obesity, most of which are epigenetic and environmental factors and lifestyle. But recently, researchers have found many genetic factors that might not cause the development of obesity directly, but it might be used as a target for the treatment of the disease. There are many genes which are involved in obesity during the inflammation. You can't see the title here, but it says thermogenesis, and this is adipogenesis. I'll come back to it later. And this is Nigella sativa. I'll remove it. Okay, so for targeting the genes involved in obesity, we turn to spices. India is the largest producer of one of the largest producers of spices, and we prepared an oleoresin from Nigella sativa seeds, which is also called black jeera or kalonji in North. Yeah. Nigella sativa has been proven to be a very good anti-inflammatory and antioxidant. And there are many cancer studies which have had positive effects from Nigella sativa, but there are not much data for the metabolic syndrome. So oleoresins are nothing but just the concentrated form of the semi-solid concentrated form of the spice oils, which reduces the loss of bioactive compounds and also increases their concentration in the intake, because we know that spices are good and we have been following them for many years for their pharmaceutical properties. But the only problem is intake. Like, we don't take spices in much amount to be effective. So here. What do I have to do? Yes. We targeted two major metabolic pathways. First is thermogenesis. This is thermogenesis. Thermogenesis is nothing but the conversion of white adipose tissue to brown adipose tissue. In brown adipose tissue, the UCP1 gene, there are more mitochondria. You can see in the picture, the brown fat cell have more mitochondria, the brown color ones, and white cell have less. So more mitochondria means more UCP1 expression. And UCP1 is the gene that reverses the proton gradient in ETC, like electron transport chain. So the energy is not stored as ATP, it's released as heat. So people with more brown fat will release their energy, like the calories they take, as heat rather than storing them in the liver and adipose tissues. So we check the expression with our product of UCP1, which I will discuss later. And the next pathway that we targeted will be adipogenesis. Adipogenesis is the generation of new adipocytes. So when there are new adipocyte formation, there's excessive lipid accumulation, which leads to hypoxia and inflammation. So we all know that yeah, I'm just done. Yeah. Okay. So there are three objectives: extraction of the spice oleoresin, the biochemical characterization, and the evaluation in vitro assays. And third will be the confirmation of anti-obesity activity in animal models, which is C57BL6 mice. This is the transgenic mice for obesity studies. So we extracted with solvent extraction. We took three solvents: methanol, ethanol, and acetonitrile, just to compare the properties. And we did different polyphenols assay, flavonoids assay, antioxidants, and in vitro enzyme inhibition assay. Three enzymes: pancreatic lipase, amylase, and glucosidase, for checking the glucose metabolism and lipid metabolism. And in animal studies, I divided animals into 6 groups. One was given normal diet, and the rest was given high-fat diet. One in the high-fat diet, we have control. One is positive control, which is with the standard Olistat, and the 3 groups are different concentration of the oleoresin: 50 mg, 100 mg per kg body weight, and 200 mg per kg body weight. This is the HPLC graph. So we can see the highest peak of thymoquinone. Thymoquinone is a major bioactive compound present in the Nigella sativa. So we can see it's present in all the three samples, and that is the standard, the fourth one. This is the GCMS for these are the list of all the polyphenols and essential fatty acids that are present in the oil. This is the result for polyphenols, flavonoids, and radical scavenging activity. We can see the more than 50% activity, antioxidant activity here. Okay, this is for anti-obesity and anti-diabetic enzyme assays. Here also we can see more than 50% in enzyme inhibition. And these are the body weight changes. So here we can see that the treatment groups have less body weight by the end of 8 weeks, rather as compared to the HFD group, which is the red line on the top. These are the serum cholesterols, triglycerides, HDL, and LDL levels. I'll summarize this. In the treatment groups, we saw best results in the 50 mg per kg body weight group and somewhat in 200 mg per kg body weight group. We were also like shocked because we usually expect concentration dependent effect, but here we saw 50 mg per kg body weight giving better results. Okay, these are for the RT-PCR results for the genes that we did. These are the adipogenesis genes, PPAR, gamma, AP2, leptin, and adiponectin. All of these genes mostly increase in high-fat diet groups, and we can see that, the second bar. And these are the thermogenesis genes, which increases the brown adipose tissue number. So UCP1, COX-8B, and PRDM16, which are all increased in the treatment group as compared to the HFD group. These are the liver sections and adipose tissue sections. From the arrow, you can see the more vacuolization in the liver, like the different morphology. And in adipose tissue, you can see the like the crown formation. Like these are not normal as compared to the normal slide. In conclusion, we can say that spice oils can be used as the treatment, or like not the treatment, but the better management for the anti-obesity or anti-metabolic disorders. And there are many challenges that we are still having. We are thinking of making the nano formulations of the spice oils to see the better effect in bioavailability. And we also want to see other genes such as inflammatory genes. And we also want to take the compound to clinical trial. And we want to make it palatable, because pungency is the better, like the more challenge for us, because it's a little pungent. It has a pungent smell. So for that, we want to make a product out of it, and we'll take it up to the clinical trial. Thank you. Yeah. 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 Yeah, yes, ma'am. Actually, yeah, this product is natural. So main problem is bioavailability. So we're thinking that uh, like it's more bioavailable in lesser concentration. No, 100 and 200 were little similar, and 50 had a better result. So that is why we are thinking of making it into a nano formulation. So we'll we'll make it into a more smaller concentration, and then we'll increase the bioavailability of it.
My one query is there.
Yes, sir.
Since it is taken as a spice and other ingredient in food item, but most of the time we are taking in a very lesser amount.
Yeah.
And if we are isolating one of the component and we are giving it in a larger quantity, have you gone through the toxic studies that it may affect the body?
The toxicity study.
Yes.
Yes, sir. Toxicity. We are yet to do toxicity and bioavailability study for the same compound in the animals only, in the mice. But since it's a natural compound, it's a spice, so we are thinking that it will not be toxic for.
All the natural components can be toxic after a certain level.
Yes, of course. And that dosage, yes.
Since most of the things we are taking as a spice only, or as a minor component in food items.
Yeah.
And if we are isolating one of the component, and you are giving in a larger quantity,
Yeah.
so that may become toxic. So that you have to study
Yes, sir. Yes, sir. Definitely.
before recommending that it should be taken for.
Yeah, that is the challenge.
obesity.
Yeah. Yes, sir. Before that, we have to do the toxicity study first. Thank you.
Thank you, Isha. I would like to call Ashutosh Mahapatra.
A very good afternoon to all present over here, all my dear colleagues, respected faculty members, and all the students to technologists. I hope you are enjoying the AMIFOST conference. So, as a part of my PhD research work, the title of the presentation is Targeted Control Release and Behavioral Kinetics of Spray-Dried Probiotic Lactobacillus acidophilus in conjunction with artificial neural network and genetic algorithm. So, before going to the topic, just I'll give a brief statement like why we are conducting these studies. We are taking a we are tying up with the aviation ministry, and we are trying, like, when we are talking about probiotic, obviously probiotic everyone knows. So we are enriching the dairy based products with probiotics in a form of capsule, so that it can be mostly used by the defense people, the first target, and like people who defense person who are mostly using staying at the high altitude. For them, it will be definitely good product. And second, when we are traveling through aeroplane, for example, we are consuming curd. Mostly, if a curd of 100 gram and 180 passengers are there, so total 18 kg of total load is there in that aeroplane. So we are trying to substitute that curd in the form of a capsule, so that the whole with a high probiotic amount, so that it will be very much beneficial for the people as well as the normal persons also. So based on that target goal, the objective of the present study is to optimize the wall material for the encapsulation of probiotic acidophilus using spray drying technique in conjunction with ANN and GA. And the second objective is to study the behavioral kinetics with targeted controlled release of L acidophilus during its biochemical characterization. So for the optimization purpose, a 3-variable 5-factorial CCRD design has been adopted. The spray drying conditions are you can see 150 degrees Celsius and 2 ml per minute, and 3 types of wall material has been selected, that is skimmill powder, maltodextrin, and cellulose acetate phthalate with a different different range. And the experimental design, this is the experimental design with different trials of run. 5 response variable has been studied, that is survival rate, log reduction, encapsulation efficiency, moisture content of the powder, and the total recovery yield of the spray dry. So, it from the response survey plots, it has been clearly found out that all the wall material, that is a ternary wall material matrix, that 3 wall materials have significant effect on all of the responses. So when we are trying to optimize, there are like several optimization methods are there. RSM, there mostly now people are using RSM. So with RSM has some certain limitations, and to overcome that limitation and to increase the efficiency of the overall optimization technique, ANN has been used. So here, like you can see, there are 3 types of input parameters are there. That is the wall material, independent variable, and 5 response variables have been studied with different iterations using the particular coded algorithm. So here you can see at the ANN can efficiently predict the whole model. It can train the data, validate, test, and the overall R-square value are quite getting high, like 0.99746. And for the second iteration, that is when we are talking about genetic algorithm or artificial neural network, it comprises of several iterations, like different type of combinations it will try and it will give a best result. So in the second iteration, that we are giving the better result. These are the predicted value and actual value for all of the responses using ANN.
and these are the relative errors. For the optimization purpose, we are using genetic algorithm. So both are the two different terms. ANN is used for the prediction of the response variable, and genetic algorithm is used to optimize the independent parameters. So in our context, like first three independent response variable, that is survival rate, encapsulation efficiency, and recovery yield. The goal is to maximize these particular three responses, and log reduction and moisture content needs to be minimized for the powder. So these fitness equations are using these particular equations, which has been generated in RSM. These equations have been fitted in the MATLAB, and the best optimized conditions are found that skim milk powder at 10% weight by weight, maltodextrin at 11.281, and cellulose acetate phthalate at 2.577 weight by weight. And here the total emulsion has been made as 30% TS. Based on the preliminary studies, the total TS is kept 30%, and in 30%, these are the optimized conditions. And these are the experimental and predicted values for the responses. Coming to the biochemical characterization of the encapsulated lactobacillus acidophilus powder, mostly in vitro studies has been done, like viability of acidophilus against simulated gastric fluid, simulated intestinal fluid, and different pHs. Because when you are consuming a food product and when it reaches through the intestine, it goes through the food pipe, stomach, small intestine, large intestine. There is a maximum chance that most of the bacteria will get died, because due to the harsh environmental conditions, low pH conditions, most of the bacteria used to be getting died. So in keeping this thing in the context, the data has been found out that the optimized encapsulated powder is sufficient enough or efficient enough to keep the core material, that is acidophilus, probiotic acidophilus, at high survival rate. When we are talking about probiotic consumption, the minimum amount of consumption we need to take is 10 to the power 7 or 7 log CFU. So in the graph you can see for the control sample, the at minimum 7 log reduction has been found out. However, for the optimized powder sample, almost 2.5 log cycle reduction is found out in salivary phase, gastric phase, and intestinal phase. That is the whole digestion process continuously. Yeah. So, like from the same graph, like all the particles are spherical in nature. I'll just take a minute. So for some spherical type encapsulated particles, there are different type of models which can be used to check the behavior or its kinetics. So these are the couple of 6 models which have been like mostly in drug release case, this type of models has been used. So after fitting, it has been found out that for simulated gastric fluid, simulated intestinal fluid, for pH3 and pH4, the type of release mechanism is supercase 2 type transport, and for simulated intestinal fluid, pH2, the type of transport or release mechanism is anomalous transport. So what these type of transports are there? Basically, there are several number of transport mechanisms are there, in which governs the mechanism and how the core metal is getting released from the wall material. So in our case, anomalous type transport symbolizes the erosion control release and diffusion control release. So if we are subjecting the wall powder in pH2, then the governing mechanisms will be erosion based control release and diffusion based control release. If we will be subjecting to other SGF, SIF, and pH3 and 4, the supercase type transport which has been found out that governs by erosion, swelling of the relaxation of the polymeric chain, and all the stresses involved. These are the couple of mechanisms which can be which is the main governing principle in the intestine or in the stomach, how the core metal is getting out of the wall metal. This kind of studies, these kind of studies are really much helpful to understand the mechanism, or if you are taking any type of medicines or capsules or any nutraceutical foods, how it behaves inside the human GIT. Using this understanding, the final later stage of my research, that is, we are making a capsule or powder which will dip in the water. It will form curd. All the necessary ingredients to make a curd with high amount of probiotic bacteria will be there alive in that dried form of powder. And whenever we require or we want to have curd, we will just put the capsule in the water some recommended amount, and it will form instant curd with high probiotic benefits. So that's the sum of my research. Thank you so much. Yes, sir. Like for now, we are after talking to the industries, we are thinking to give the powder in the form of LDP in the first phase, because the overall cost is getting high from now, right now. Although we are using the spray drying, but using all these wall materials, cellulose sintered phthalate, and the overall technique, the cost is quite getting high. So in yes, sir. So initially, we are recommending like to the defense people, once the things will be getting accepted, then we'll try to minimize the cost. So in the initial phase, using LDP, we are trying to package the whole. Negative encapsulation. Just a second.
I think previous one.
This is the efficient encapsulation efficiency, and negative, that is the relative error. I guess that is relative. That is the relative error between the predicted value and the calculated value. Like software will give a predicted value, and when we will use that particular optimized conditions in the real experimental condition, we'll get some of the responses. Like software will give the encapsulation efficiency is 95%. When we are calculating on the real experiment, we are getting, for example, 94%. In that context, the relative error will be -1.
Okay.
Because it will the actual value minus the predicted value divided by the.
So it is given by the RSM only.
It can be normally calculated.
Normally calculated.
Here, this data has been found out from the ANN.
Okay.
But it can, once you got the predicted value, you can only do the predicted value. Once you got the predicted value, you need to do on the real experiment. Then you can calculate the relative error.
Okay. Thank you. Thank you.
Yes. Yeah. Yeah. Actually, here in India, we can use either from the bacteria which I procured from the NDRA kernel. So either MTCC strain we can use or ATCC strain we can use. This is the standardized strains which can be used in India.
So ATCC, that 11975 strain is
that is the particular species of lactobacillus which has grown in India. They have cultured it, and it is the standard thing. So we just procured from that culture from NDRA kernel in the live form. Like for example, we are procuring a raw material. Similarly, we can procure the particular type of strain from the.
Because there are a lot of strain differences in the way the curd is prepared.
Yes,
so why you have taken that?
For
and why not you have...
acidophilus, NDRA has grown this particular strain. Okay, so that we are. Thank you.
Thank you, Ashutosh. So I would like to call Mr. Neeraj Srivastava for his presentation. Next, Nishi Singh.
Shall I start? Okay. The voice of the afternoon to the respected designatories and the members chairing the session and the participants. I am Nishi Singh and I'm a PhD research scholar from National Dairy Research Institute, Karnal. And today, through this platform of 6th AMIFOST, I am supposed to put forward my oral presentation on the topic entitled as the study of migration behavior of plasticizers and antioxidants from the LDPE and LLDPE films into the milk. So here's the general flow of my presentation, starting with the general introductory slides, followed by the broad research objectives, and thereafter the methodologies adopted to conclude with the results and the findings, and with the due acknowledgements, I'll end my presentation. So to begin with, here is the general illustration which states that how the packaging integrity of a packaged food would be compromised if the hermetic sealing, or say about the delamination properties, fails, or rather the unification of the packaging material as a whole, it fails against the ingress of the unwanted gases, or say about the water vapors and even the aroma. Parallelly, when it is the chemical migration of the chemical additives which is happening, it in all together with the residual monomers as well as the impurities will impede, or say about compromise the safety assurance of the packaging material. In yet another depiction here, it says that chemical migration is basically a diffusion phenomena in which the chemical additives which are added to the packaging material during the compounding stage, they migrate, or rather they diffuse from the packaging material into the food, which is in an intimate contact with it, and which obviously have negative human health implications. So now in yet another slide, so here is the rationale which states that why specifically the milk as a commodity has been chosen. So here is a reflective of that how big is the Indian dairy industry, which says that India is one of the world's largest producer with the 221 MMT of its production, followed by only 25% of the total produce enters into the commercial value chain, and of which 64% is directly manufactured and marketed as the pasteurized milk product. And this packaging material is a co-extruded multi-layer flexible pouches, which is made out of LDPE and LLDPE blend, and which is very elusive to India. You are not going to find it anywhere else on the globe, it says. And so now here are the broad objectives, which says that it is a 3-tier research study in which initially the chemical additives from the packaging materials were exhaustively extracted using the focused ultrasound liquid solid extraction. And it was followed by that we basically targeted the 10 frequently migrating components, or say about the plasticizers and the antioxidants. And then a migration was traced in the milk as a biomatrix. And thereafter, the analytical protocol, which was developed for the quantification of these additives, they were validated. And thereafter, the mathematical modeling for the predictive migration was also done. So to begin with, here's a methodology in which the packaging material, which was comminuted, and it was again in intimate contact with the extraction solvent and the focused ultrasound liquid solid extraction microprobe, and thereafter it was extracted at these 3 different parameters, and due to the cavitation effect, the additives were extracted. And these this is a heat matrix which shows that the total iron concentration of 62 different types of components which have been extracted using the pulse technology, and of which only these 10 which were frequently occurring, as well as they were migrating in one of the highest quantities, they were put forward for tracing their migration
into the milk. And this is like the illustrative GCMSMS graphs, or say about the MRM optimization, where these 10 chemical additives, they were quantified as well. So thereafter, milk as a biomatrix, from which these 10 chemical additives were extracted using the liquid extraction technology, which was based out of low temperature. And the study of migration kinetics was done from 0 to 48 hours, which is the shelf life of the milk under refrigerated condition at two different temperatures. One was 4 degrees centigrade and the other was the ambient temperature, which was again at 25 degrees centigrade. So these are the validation study that these 10 chemical additives, they were validated, and in terms of the linearity, percent recovery, even the LOD and the LEQ. So what I can claim through my analytical procedure, which was developed, that it can the liquid extraction based low temperature protocol can detect the chemical additive which has migrated in the milk as low as 0.02 ppm. And this is the concentration of the 10 different chemical additives which have been extracted from the milk. And then to understand the behavior of the different chemical additives and what are the different factors which are affecting the process of diffusion or the migration. So there were 3 different models which were studied. The initial 2, the FDA and Brandeis equation, these are like something which has been endorsed by US FDA and EU regulation as a whole. And since our packaging material is elusive to India, so we have tried Wibble model as one of the elusive model here. So by using FDA and Brandeis equation, we came to conclude that smaller is the molecular weight of the chemical additives, more frequently and more easily they're diffusing into the food matrix. And again, the thickness of the packaging material is also causing a difference, followed by if the initial concentration of the chemical additives is higher, so there are higher chances of it migrating into the food matrix. So as you can see that the diffusion coefficient, which is depicted here as DP, using the FDA and the Brandeis equation, has been found as 2 different digital values. So which says that FDA as well as the Brandeis equation, they predict in 2 different forms. And thereafter, another factor which contributes to the diffusion of the chemical additives from the packaging material is the temperature of the exposure, which provides the intimate contact with the food matrix as well as the chemical additives which are migrating from the packaging material. And thereafter, in Wibble model, there were 2 crucial parameters which were calculated, and the Wibble model was something which gave the concentration of the chemical additives rel to that of the experimental values. And this is how it is illustrated in all these graphs. The red one, which says that it is a Wibble fitting, which using the Wibble model we have predicted, and through GCMSMS, the black dot, which says that it was the experimental values. So the R-square values of the goodness of the fit, which was received using the Wibble model of fitting, it says that it was below 90, which is the best fit model. So quick takeaways from my research findings, it says that the migration of the quantified chemical additives in the milk are not as alarming, and there have been methodologies which have been developed and validated, and it has been now in the draft phase, and the FSSAI is yet to adopt it. And Wibble model is something, one of the best predictive modeling which we have proved through our findings that it best fits the experimental data. And now the due acknowledgements to my principal institute, NDRI, and Ministry of Food Processing of India for financially funding it, and ICMR for financially funding my research journey throughout, and our industry partners, IDMC and Uplex India. And that was my time on the dais. Jai Hind. No, ma'am, it is just about the overall migration, which is like, say, about the objective safety of the packaging material, it will say. But they are not specifically telling that what quantity of the plasticizer antioxidant are allowed to be, yes, the specific migration limits have not been regulated in India so far. So I guess out of our research publication, they have adopted 2, and because it was a financial funding from ministry. So now we are yet in draft. Thank you, ma'am. Thank you.
audio. This one. A very blissful good afternoon to everyone. I'm Asta Diwan, Assistant Professor in the Department of Food Technology, Guru Jamdeshwar University, Science and Technology, Hisar. Today I'm here to talk on impact of gluten fraction on the microstructure of the dough. So, to talk about, just I'll give a brief. Food microstructure is essential to know the impact of the processing parameters, influence of the ingredients and their interactions. So that's why we go for the evaluation of the microstructure. And to know the gluten network impact on the microstructure, this could be a substantial step towards improving the flour industrial efficiency and establishing the functional role of the individual fractions of gluten on the dough rheology and at last the end product quality. So we know that gluten, it is a wheat protein which is combined when gliadin and glutenin are hydrated and kneaded to form a dough. So gluten is developed. They have a mesh viscoelastic-like structure. So many studies have been done, and they have studied the microstructure of the flour dough, but not with the isolated fractions. So here is the labeling that you might heard from me, hear from me, that what kind of terms I'm using to differentiate the effect of addition of that particular fraction. That is sheet-like gluten structure, protein discontinuity, the globules which you will see in the micrograph will be can be recognized as starch granules, and the thin gluten sheet films kind of structure. So what methodology I adopted? I isolated gliadins and glutenins from the wheat flour, employing Osborne fractionation. 70% of 70% ethanol was used for the isolation of gliadins. Solubles were gliadins, and insoluble were recognized as glutenin. And these were my fractions. Then I isolated the HMW and LMW. These are the fractions, subfractions of glutenin, polymeric wheat protein. They are being subfractionated into high molecular weight glutenin and low molecular weight glutenin. So I adopted the method of Millar's 1994, which was acetone precipitation method, in which when we add 40% acetone, the HMW will be precipitated, and the when we add 80% acetone, LMW will be precipitated. So these are the fractions. I
prepared the dose samples like this. Fortified dose samples were prepared and they were exposed for the gold coating and scanned at 2x. So these are the results. To have a specific look, I would like to say, here you can see when the glidines were added at the rate of 3%, there is varietal variation. In DBW, natively, more of the glutenin percentage was present. When glidines were added, you can see the opening of the structure, means more vacuoles can be seen. But when it was added in variety HI 1500, it was natively composed of higher percentage of the glidines. So a sheet-like structure was formed. So when the glutenin were added, in HI 1500, it was natively composed of less of the glutenin. So you can see some strands which are being increased, but still there is vacuoles in the structure. But when it was added in DBW88, it is a strong variety. It was natively composed of the higher percentage of the glutenin proportion. So a sheet-like, a dense and compact structure was created. So conclusion was that when we add glidines, it leads to the opening of the dough network. But when we add the glutenin in stronger variety, it makes the dough more stronger. But when we add the glutenins to the weak variety, it leads to a strength, but not to that level. When which it can create in hard variety, strong variety. So with respect to the evaluation of HMW and LMW addition in both the varieties, it was observed that when HMW is being added in the strong variety, more dense and compact structure is being created because it acts as a backbone for creating the dough gluten network structure. But when it was added in HI 1500, small vacuoles or cavities or gaps we can observe in the micrograph. To have a clear impact, you can see there is in HI 1500, there are gaps or cavities in the microstructure, but in DBW 88 with 5% of HMW, there is compact, dense dough structure is being created in the gluten network. When we talk about the LMW and of both the varieties, it was contrasting that LMW of DBW 88. It is a strong variety. It shows the less strength on comparing with the LMW of the HI 1500. So you can see the cavities in the first image rather than in the second image. So it was overall concluded that a varietal composition variation was observed in the microstructure of the dough when it is being supplemented with their isolated fraction. So individual fractions of the gluten revealed its individual functionality in the dough microstructure, and it was determined through SIMS, scanning electron microscope. So that's all. You can ask for the queries. Any query? Thank you. Thank you for giving this opportunity. Thank you.
Thank you, Asta. So I would like to inform all the participants that the second session has been started, and I would like to call Tarubi.
A very good afternoon to everyone. Myself, Tarubi Agrawal, Assistant Professor in Government Degree College and got selection through UP-PCS. So I'm today going to present a review paper on food-based strategies to combat micronutrient malnutrition, a narrative review paper. So as we know, malnutrition refers to the deficiencies, excess or imbalance in a person's intake of energy or nutrients. The term malnutrition addresses three broad categories: undernutrition, micronutrient malnutrition, and the overweight. Now, what is micronutrient related malnutrition? Inadequate intake of vitamins and minerals, often referred to as micronutrient malnutrition, and iodine, vitamin A, and iron are the commonly prevalent deficiencies globally among the pregnant women and children in low income group countries. Why this topic I have selected? Because despite several government initiatives, more than 2 billion deaths occur from micronutrient malnourishment, and this might be due to lack of knowledge and lack of commitment that were identified as a major drawbacks in an effective implementation of evidence-based policies that address dietary deficits. Moreover, micronutrient deficiencies negatively impacts the intellectual and physical health of the children, potential of the children. Thus, looking towards the significance of the situation, the present review paper aimed to address the food-based strategies that combat the micronutrient malnourishment. So, according to FAO, food-based strategies encompasses a wide variety of interventions that aim to increase the production of micronutrient-rich foods, increasing the intake of micronutrient-rich foods, and increasing the bioavailability of micronutrients. In this context, the need for food fortification, the food supplements, and diversification is highlighted. So, what is food supplementation? Food supplementation is defined as any food which aims to supplement the normal diet, and nowadays it is available in the market in the form of capsules, gummies, powders, tablets, and popular supplements include vitamin D, vitamin B12, and in the minerals, calcium and iron. Food fortification is scientifically proven, cost-effective, scalable, and sustainable global intervention that address the issue of micronutrient deficiencies, and commonly staple fortified foods are wheat flour and rice with iron, vitamin B12 and folic acid, milk and edible oil with vitamins A and D, and double fortified salt with iodine and iron. Dietary diversification or modification, it involves the changes in the food production changes, food selection patterns, and traditional household methods for preparing and processing indigenous foods that aims to enhance the availability, access, and utilization of foods with a high content and bioavailability of micronutrients. And some other strategies to enhance the content or viability of micronutrients in plant-based foods are field fortifications, genetic engineering, and plant breeding. So, future research, what is the recommendation of this review paper? The future research is needed to develop national nutrition programs with the involvement and education of all stakeholders, particularly policymakers. Thank you to all. Thank you, ma'am.
Thank you, Tarubi. I would like to call Miss Ankita Vare.
A very good afternoon to all. Thank you for giving me the stage to talk on my to discuss on my topic of utilization of in vitro digestion model to check the efficiency of herbal nanoemulsion for lipid digestion process. My name is Ankita Vishwanath Avari, and I am a PhD scholar from lovely Professional University, Punjab. So for introduction, we know that the world is battling with obesity right now, and it has been a growing concern. So when we talk about lipids, and one of the main component or the factor responsible for obesity is called the lipids. Lipids are known as the basic components of a cell membrane, and they play an important part in human health. Lipid metabolism includes, when we talk about their functions in the body, they include the biosynthesis and degradation of the lipids. And statistically, 38.2 million kids under the age of 5 have been reportedly overweight or are obese in the year of 2019. And the prevalence of obesity in India is about 40.3%. Now, when we talk about medicinal plant as a source of medicine, so India is known as the land of Ayurveda, and this is made possible due to its different availability of Western Ghats or Eastern Ghats, and they have numerous plants which can be used as a herbal medicine. So why do we need, so according to WHO estimate up to 4 billion people or 80% of the world population are still relies on herbal medicines. And this is due to the fact that they're affordable, they do not have any side effects, and they can be long lasting. And when we compare it to the allopathic medicines, these medicines target the root cause of the problem and not just the surfaces. But the only disadvantages it has is it has an increased systematic clearance. That means it has, as soon as we intake these medicines, the chances of it being removed from the body is higher. And therefore, it needed a repeated administration, which can sometimes lead to toxicity. Now, herbal nanoemulsion. So when we talk about nanoemulsions are colloidal particle systems, and they are of submicron size, which ranges between 20 to 200 nanometers. And this is, when we talk about nanoemulsions, we can talk about them as a shell which covers our drug, and it can be used to administer our drug into the human body. Now, the advantages of using a nanoemulsion in medicine is it can be used to dissolve huge quantities of poorly soluble drugs, and it can be used to extend the release of the drug. And there are more routes for delivery of drugs are available because of the nanoemulsion, and it can be made, or it can be used to have a target release in the body. And due to the high surface area, it can be easily absorbed by the body cells. So in vitro, what is in vitro digestion model? So when we talk about in vitro, ma'am, actually my presentation is more review based. Yeah, because I've just started my work. So when we talk about in vitro digestion model is just creating a model, converting an in vivo human digestive model into an in vitro digestion model where we can easily study the different effects and have an uniform results. So for as a plan of action for my project, we have selected 6 herbs which are present in which are indigenous to Western Ghats. And then from there, using a deep eutatic solvent method, we are extracting the main components which act as an anti-lipid. And then we create nanoemulsion formulations, and these nanoemulsion formulations are studied for their activity to digest lipids. And these studies are then the selected nanoemulsion are then put into in vitro digestion studies, and their actions on the body is studied. And at the same time, we also study the shelf life of the nanoemulsions. So, are there any questions? Thank you so much.
I would like to call Sneha.
Good afternoon. I'm Sneha, PhD scholar from Lovely Professional University. My research is on the effect of germination of pearl millet
at different temperature and for different time period on antioxidant activity, total phenolic content and tannin content. So pearl millet is one of the most common staple crop in India. It is also more highly consumed worldwide. The most prominent limitation in pearl millet is the presence of anti-nutritional factors such as tannin and phytates. But processing of these millets can reduce the tannin content, and also to see that the germination does not affect the other important components like the phenolic components or the antioxidant activity. The objective of the study is to study the effect of germination on pearl millet grains at different temperatures and time, and the effect of germination on antioxidant activity, tannin, and total phenolic content. So this is the overall of the germination condition that was used. The grains are soaked for 16 hours at 25 degrees, and then germinated at 22, 26, and 30 degrees for 24, 48, and 72 hours. And it was again dried at 6 degrees Celsius in tray dryer and ground into powder before actually taking them to further analysis. So after the germination, we check the shoot length of the seed, grain in millimeter, and check the total phenolic content by following CO-Cal2 method, antioxidant by DBPH inhibition percentage, and tannin content by Faunen-Dennis method. So according to this, the highest growth was observed at temperature above at 30 degrees Celsius, and the growth was highest after 72 degrees, after 72 hours, and which was comparable to the growth at 72 after 72 hours at 26 degrees Celsius. The total phenolic content seems to be increasing. This is probably because most of the phenolic components are isolated on the seed coat, and which is not very much involved in the germination process itself. So, like during the process of germination, because of the decrease in overall mass of the germ, the phenolic content seem to be increasing. So in DPPH, the percentage inhibition seem to be reducing in spite of the phenolic content seem to be increasing. This could be possibly because of the decrease of other non-phenolic components such as phytic acid, which also contributes to DPPHSA. The tannin content also seemed to be increasing, but at 30 degrees, the increase was much lesser than at 26 degrees Celsius. That's all. No, ma'am. Just yes, ma'am. Thank you.
Thank you, Sneha. I would like to call Mr. Bittu.
Good afternoon, everyone. I am Bittu Prithvi Kumar, pursuing MTech second year from IIT Roorkee, and my sincere thanks to Kirtiraj Gaikwad sir. My topic is novel 3-hydroxyphenol based moisture activated oxygen scavenger for active packaging. These are my contents, which I am going to cover in the further slides. Basically, main aim of the packaging is to protect its content without any damage. Some of the objectives of packaging are product promotion, convenience, product identification, and protection. Active packaging systems. Active packaging systems helps in extending shelf life, monitor freshness, improve safety and convenience. These are some of the active packaging agents like oxygen scavengers, ethylene scavengers, antimicrobial, carbon dioxide emitters, absorbers, antioxidants, flavor releasers, and absorbers, moisture regulator. These are incorporated inside the package in order to reduce the spoilage of foods or pharmaceuticals. In the presence of oxygen in the food can cause off-flavor development and nutrient loss. So oxygen scavengers are incorporated inside the package in the form of sachets or films or labels. This is the detailed classification of oxygen scavengers, like classified into metallic, organic, inorganic, and polymer-based and enzyme-based agents. My main research objectives is to develop a novel-based oxygen scavenging material by using a 3-hydroxyphenyl as an active material and evaluating oxygen scavenging capacity at different temperatures maintained at different humidities, and to study the effect of temperatures and humidities on oxygen scavenging capacity. After that, studying oxygen absorption mechanism by using kinetic curve fitting model. In this, evaluation of oxygen capacity and absorption capacity and rate at different temperatures and humidities are calculated and develop comparing the rate constants for different active materials. This is my active material, 3-hydroxyphenol. It is also known as resorcinol or meta-dihydroxybenzene. It can oxidize in presence of alkaline substance or metallic catalyst. It has FDA approval and it is generally recognized as safe material. It is suitable for coating applications on paper substrate. This is the chemical structure of resorcinol. Here are the experimental details. Firstly, resorcinol and potassium carbonate, which are GRAS approved ingredients. Firstly, sample preparation has been done, which is poured inside a 100 ml glass vial. After that, crimp sealing has to be done in order to not to enter the air inside the glass vial. After that, by using the 3 gas phase analyzer, we are calculating the O2 concentration. After that, data validation is to be done. After that, this is the experimental composition of my experiment. The RC sample code is RCPC 1:0, which can be named as control. Where I have taken 1 gram of resorcinol and 0 grams of potassium. Similarly, like RCPC 1:0.25, 1:0.5, 1:1. Coming to the results and discussion, the diverse colorimetry, the diverse oxygen system subjected to various temperatures and humidities can be better distinguished by their colors. Like we can see before scavenging, it is white in color, and after scavenging, like brownish red in color. So by using the formula delta E, total color difference, we can be calculated like delta E. And this is the oxygen scavenging analysis. We can clearly see we are subjected the glass vials to different temperatures, maintaining at different humidities. Like firstly, we have maintained at 25 degrees, maintaining different humidities 11%, 75, and 99% RH. The oxygen content decreases from 20.9 to 0% at 99 degrees. At 99% RH, it decreases very fast as compared to 75% and 11% RH. And at 45 degrees, maintaining at different humidities, it decreases even faster as compared at 25 degrees centigrade. As at accelerated temperature, molecules tends to react faster, so the oxygen tends to decrease faster as compared to at 25 degrees centigrade. Coming at 5 degrees centigrade, maintaining at different humidities, like at 11% RH, the oxygen content not decreases. It maintains constant at 20.9. At 75% and 99%, except the control sample, remaining samples tends to decreases from 20.9 to 0% after the time we have. Our foods can be better protected, can be stored in refrigerator except at 11% RH. We can store at 75% and 99% RH. This is the effect of storage temperature and humidity on oxygen scavenging capacity. By using the formula rate, oxygen scavenging rate, we can calculate oxygen scavenging rate that into multiplying with time we can get oxygen scavenging capacity. So for different samples, I have noted at a different temperature oxygen scavenging capacity and oxygen scavenging rate. As the temperature increase, we can see from the graph, like as the temperature increases, like oxygen scavenging capacity also increases at the different humidities. And coming to the effect of storage temperature and humidity on chemical kinetics, by using kinetic curve fitting model, I have calculated rate constant values for my non-metallic based oxygen scavenger, maintaining at different temperatures at different humidities. So as the temperature increases, like the rate constant also increases, rate constant values. And comparing comparing with the different rate constant values, like non-metallic based agent, fresh packs, iron based, enzyme based, sodium erythrobate, and natural rubber latex based. Our rate constant values as compared to different commercial ones, it is higher. So this can be a good alternative compared. Our non-metallic based oxygen scavenger can be a better alternative as compared to commercial one. Thank you. Like by using 3 gas phase analyzer. Like we can get, firstly we need to calibrate the oxygen present in the air. After that, at different times, I have measured by using that gas phase analyzer. Thank you.
Lokman Hakim.
Good afternoon, all. Myself Lukman Hakim. I am doing masters in IIT Roorkee, and I'm working under Professor Kitiras K. Gaikwad. And my project is the development of novel edible ink for food printing and packaging application. Here's my content. So let's talk about the commercial printing ink. We all know that commercial printing ink is a petroleum based, and it is a chemical based ink which is non-renewable, and it has very people are using industries are using toluene, that is toxic chemicals. So you also see these kind of stickers in fruits and vegetables. So basically the purpose of this stickers to like if you see some shop, one is sticker on apple, another is without sticker. So we usually think that the sticker on is premium, it's the quality is good, but they use the purpose basically use to traceability, and like sometimes they use for the hide the decay and the defects. So now question may arise, like, are they safe? Like, because these kind of fruits are porous in nature, and this kind of sticker they apply to the pressure sensitive adhesive, and again sometimes accidentally children they consume this kind of sticker. And how do? We peel off the, when you buy this fruit, we peel off the sticker and slice, and then after that we eat. And you all see this kind of base like in the sweet and like laddu, something like that. Actually, this contain the chemical based ink. Sometimes accidentally we consume this kind of sticker base. Otherwise, this ink migrate to the food. Again, you see the third picture. That is the sometimes if we buy the street food, then we use the newspaper. Again, the newspaper in the newspaper, there is a chemical based ink that is migrate to the food. And nowadays, the photo cakes in the birthday, and it is very famous. But again, and this cakes, personalized printing or something like that, that is again, they are using synthetic ink. That is again, yeah, we can eat, but again, it is synthetic. And here I have mentioned the migration, 3 type of migration. So the problem statement was the first migration, toxicity, biodegradability, and the cost. Cost is the major issue. I'll discuss in the upcoming slide. So for that, we have developed the edible ink. For that, we have taken the ketachu as a coloring compound. And we all know that ketachu is a natural bound dye that is green alternative. And it has traditional, like ketachu used in traditional medicines also. It has a lot of benefits like antimicrobial, antioxidant, and antibacterial, antifungal, and so on. We have used guar gum as a natural binder that has also multiple benefits that provide also sharply printed patterns, constant high viscosity, and so on. So our objective was to develop an edible environmental friendly and edible ink by using plant source to evaluate the coloring properties of ketachu, plant-based materials, and so on. So here is the technology development. First of all, I extract the ketachu, and I have collected the wood source, and we make it, we chips it into small pieces. We boil it, and after that we filter it, we evaporate it, and freeze it minus 70 degrees Celsius, and we got the powder. So after that we prepare the edible ink. For that we have used the coloring solution, additive solvents, moderant, and binder. We mixed it, all the components, and we have reduced the particle size by the ball milling. Finally, we got the edible product. So I have used the printing method, that is screen printing method. So I have, first of all, I have created the with the IITR logo, the stencil, and I have printed on. This is the I have printed on paper, different paper. I have also printed on like pizza base bun. Nowadays, you can see that there is an egg with printed something karaknath egg. There is expensive egg. So we are seeing the printed egg. Again, the problem is that the commercial ink that migrate to the food. So we are using the edible ink. We can also print. We also print in the cookies, bread. So like I have printed on green apple, mangoes, sunmelon, on it to the stickers, and different kind of vegetables. So nowadays you see the edible cups and edible plates. Like you can after the having meal, you can eat the cups and as well as plate also. Even edible spoon also there. So, but problem is that we don't see any branding in that edible cups and edible plates. So with the help of our ink, we can print on that edible cups and edible plates. So we can consume the cups and plates as well as the ink also. So we have measured the color properties and solid content. We have also measured the rub test. That was okay. We have did light fastness also. There is the rheology properties, and in the cost analysis, the commercial ink, it has like more than 1 kg of commercial ink. It costs more than 10,000 rupees. But with the like our 1 kg of edible ink, it will cost only 177 rupees. So we are saving more than 56 times. Again, we can, if you see the stickers, to print 1,000 apple, it will cost 100 rupees approximately. And if we print 1,000 apple with the our edible ink, then it will cost only 1.77 rupees. So here is the global market of edible ink. and here is the references I have collected, and I have filed the patent also in that, and I have published the paper also. Thank you. Katachu. Yeah, actually, it doesn't have any test. It doesn't change any test. We have tested, like we have given to the mass also. We have tested with different people, around 50 people. They have eaten, and there is no change of taste. Yeah, actually, we know that the life cycle of the fruits is maximum 15 days or 20 days, but it sustains more than that, 1 month, more than 1 month. Yeah. Thank you.
Our next presenter is Miss Ranjana Acharya.
Good afternoon, everyone. I am Ranjana Acharya, PhD research scholar from Jigapant University of Agriculture and Technology. I am going to give my presentation on nutritional evolution of mushroom varieties and pre-treatment standardization for the preparation of mushroom powder. Here, I have two objectives. That is the nutritional evolution of mushroom varieties. 4 mushroom varieties are included. That is the button mushroom, desi mushroom or Ganoderma, milky mushroom, and oyster mushroom. And then after that, I have standardized the pre-treatment for the preparation of mushroom powder. As we know, mushroom is considered as a superfood because of its varieties of nutritional content, like it consists of carbohydrate, fat, moisture, ash, protein, minerals, vitamins. Along with this, it contains antioxidant compounds that have antioxidant capacities and bioactive compounds. The mushroom production in India is 0.13 million tons, whereas the per capita consumption of mushroom is only 90 grams. The material and methods include I have procured 4 varieties of mushroom. Then I have done trade drying of mushroom at 50 degrees Celsius up to the moisture content of 4 to 5%. Then I have estimated the true protein, tryptophan content, total antioxidant capacity, and the total phenol content. Apart from this, for the pre-treatment purpose, I have taken the button mushroom as it is plentifully available in Uttarakhand, and I have divided into 4 categories. That is the fresh mushroom, the blanched mushroom that is blanched for 2 to 3 minutes, the asmoled mushroom that contain salt brine solution of 10%, and the blanched and KMS treated mushroom that it contain the KMS of 0.5%. Now the mushrooms were dried, and the dried mushroom were ground into a fine powder after drying using a grinder, and proximate analysis were done. On the basis of this, the true protein, tryptophan content, antioxidant capacity, and phenol content are calculated, estimated in the 4 mushroom varieties. The result shows that the true protein content is maximum in case of reishi mushroom or Ganoderma mushroom. and also high. It contains high amount of tryptophan, whereas the total antioxidant capacity is highest in case of milky mushroom, and the total phenol content is highest in case of oyster mushroom. And the effect of pre-treatment shows that the percent recovery is highest in case of untreated mushroom, and it is lowest in case of the blanched and KMS treated mushroom. Whereas the color of the mushroom powder is more available in case of the blanched and KMS treated mushroom, whereas in other three varieties, the color is little brownish. The proximate analysis of the mushroom powder shows that the mushroom powder which are blanched and KMS treated shows a good result in case of moisture, protein, crude fiber, and total ash content, whereas there is significant difference in case of the moisture, protein, and carbohydrate content of the mushroom varieties which are pre-treated in different methods, whereas there is no significant difference in crude fat, fiber, and total ash content of the mushroom varieties. Now, I would like to conclude that the true protein content and tryptophan content are highest in Desi mushroom, and it can be used as a replacement for many animal proteins. And the total antioxidant activity is highest in case of oyster mushroom, and the percent recovery is maximum in untreated mushroom. The pre-treatment with KMS solution give the better quality in terms of nutrient content. So the mushroom pre-treated with KMS solution has good potential for value addition as powder and commercialization. Thank you. Yes, ma'am. Ma'am, true protein is a protein content that after we separate the non-protein substances from the crude protein, we will get the true protein. Non-protein fractions like uric acid, urea, and ammonium nitrate like this, creatine, creatine. Ma'am, actually, it is part of my PhD research work, and I'm working on it as a mushroom as a food source for the dietary management of depression. And as we know, mushroom is highly perishable, and it has its seasonal variability also. So we can't get it in all the day of the year. So after drying, we can take it as a food and as a food supplement, and we can use it in various products like cake, cookies, and all these products so that we can use it whenever we want. Yes, ma'am. Yes, ma'am. Actually, not more research work are done in this field, but the Ganoderma rescue mushroom has proved to be beneficial for the management of depression because mostly if we see that depression can be managed through the food as if it contain antioxidant and dietary fiber and other sources and tryptophan also. So mushroom also contain good amount of tryptophan. So I think it will be beneficial to manage the serotonin level of our body. Thank you.
Our next presenter is Miss Charu Bisht.
Good afternoon, everyone. I, Charu Bisht, student of GB Pant University of Agriculture and Technology, is going to present my preliminary trial work on the topic a transformation of spent grains into cookies, exploiting the potentiality of agro waste. As we all know, it has been predicted that the population is going to rise um, up to millions, that is around 9.8 billions by 2050. So the food demand will also increase. Due to the rise in the food demand, the food processing percentage will also enhance. And in order to meet this demand, the more the food processing, more byproducts would be produced. And more byproducts is produced, that would be very bad for our environment. So in order to minimize the agro waste has been converted into high value products, or they are utilized for the extraction of various bioactive products. Now comes the alcoholic fermentation. Alcoholic fermentation, it is the catabolic reaction in which the pyruvate is broken down into ethanol, and it is taken place under a favorable temperature, that is about 22 to 27 degrees Celsius, at a nitrogen level of 150 to 250 mg per liter. 1% alcohol production requires of about 15.7 to 18 gram per liter of sugars. And rice is also fermented. India is the second largest producer of rice. About 120 million tons rice is produced in India. Rice is also used for the production of agrochemicals. On rice, yeast cells act and ethanol is produced along with carbon dioxide and spent residue. Spent residue is the leftover that is not consumed by anyone. So, the byproduct of fermentation mainly comprises of the spent grains. Spent grains is about 85%, whereas the other waste is about 15%. Right, currently, the utilization of spent grains is not very much in human food. It is only used for animal food and for organic fertilizers, for production of organic fertilizers and for production of bricks. So, why I have taken distilled spent grains? As they possess high protein content, they possess lignocellulosic content, it has unacceptable aroma that is not accepted by human beings, it has unacceptable color, the color is very, very dark, it is spoiled due to very high moisture content, it has coarse particles, if we eat it, we feel a grittiness in our throat, and till now there is no incorporation into the human food. So, my main objective is to identify different volatile compounds present in the rice spent grains and optimization method for the removal of these volatile compounds so that they can be used in the human food. The various volatile compounds present were alcohols that gave a pungent, fermented, bready and earthy smell, aldehydes that gave a nutty, malty and caramel-like smells, and ketones that produced a floral, woody, spicy and herbaceous smell. So, various methods such as water treatment, acid washing, alkali washing were done, and independent variables were taken as time and temperature, and dependent variables that were studied were aroma by sensory analysis. The method that was optimized was soaking in water at 25 degrees Celsius for 4 hours with the change of water at every hour. By this method, the volatile compounds were reduced up to a very good level. The second objective was the size reduction and the color modification for acceptability enhancement. There is a major variation of color of these spent grains in comparison to wheat flour. So it is not accepted by human beings at all. So it can't be added to human food. So various methodologies such as size reduction was done. Second, the treatment was done with hydrogen peroxide. Then the washing in order to remove the hydrogen peroxide so that no residues is being left in the food. Then at last, the testing of hydrogen peroxide residues were done in order to confirm it.
The color analysis was done, and it was observed that wheat flour had a lightness of around 98.84, whereas the spent grains had a lightness of 48.24. So that's a huge difference. That's why they are not accepted by anyone. In the pictures, as you can see, the wheat flour, the dried distilled spent grains, and the treated spent grains. This treated spent grains has been treated with hydrogen peroxide. Now, the third objective is the incorporation of these dried distilled spent grains into human food. By these treated spent grains, cookies were made with substitution with the wheat flour. 10% substitution was done, and in that protein content was around 2.16%. 20% substitution of wheat flour was done, and the protein content was around 4.85%. At 30% substitution, the protein content was about 8.67%, and at 40% substitution, the protein content was around 10.95%. At 50% substitution, the maximum content of protein was observed, that is 14.18%. But as we see the pictures of the cookies, the 50% substitution can't be used for eating because the color is not acceptable by human beings. So still my research is going on, and I have to solve this problem. Thank you. Yeah, it's done. Yeah. Okay. So the conclusion is these agro waste cannot be considered as pollutant, whereas they can be utilized in human food so that high value added products can be produced out of these. Okay, you just
With this, we come to the end of our presentations. Now we can all move to the lunch at F3 hall. Thank you so much.